I cannot talk with this. I need to move, and this makes me really feel a bit uneasy. Okay, good afternoon again. Thank you for being here. And I don't know how much longer it's gonna take, as much as you like, it seems. Anyway, this is a round table for bees and pesticides. Usually in the program it's called pesticides, but it's bees and pesticides. We are not, we are a group of beekeepers and bee scientists, so it's bees and pesticides. And we will talk about this kind of stuff and the effect of the pesticides on bees, or the point of view of the industry. That's why in the panel we have distinguished scientists to talk to us about it, as well as representatives from industry, because we want to have opinion of both sides. And of course, also we want to have your opinion, your inputs, as well as your experience, because you might have experience if you have facing problems with your colonies. I will just go very, very briefly through the couple of points that we will try to address in very short presentations, and then give you the right to talk or ask questions. What we would like to talk about is about this controversial issue that came after the EU decision for the ban of neonics. Then it is a question for us, maybe it is for you, if we need, and if there are new tests for laboratory experiments, for field experiments, tests that they can give us more data about the effect of pesticides on bees. Which are the advanced procedures about the registration of a product? If there are any intoxication methods or mechanisms that the bee itself has, we should know about it. Maybe we can improve it. Which other stressors could have a synergetic effect with the pesticides? Is it really a global approach for this problem? Or there is different approach between EU and the States? Could we do something about it? Do we need to have, as beekeepers, a campaign for safe products? And if there are any alternatives to what we think that it is toxic. So I will not talk to you about it. I will not give you any presentation at all. It's only what you know. I have talked so much about pesticides the previous days that I have fed up. But because there are so many and very good scientists with me here, and it's for me it's a pleasure and an honor to listen to what they have to say. I will just introduce them to you first, and then we will start with the short presentations. However, because this is really a very hot and very important issue, and also for Apimondia, we have the honor to be with us, the president of Apimondia, Mr. Gilles Rotter, and I'm very happy for this as well. Apimondia would like to have an output at the end, so we might come up with a few lines to give to the outside world, saying what will be discussed and what could be the output of this round table. So I will then continue to Wolfgang Ritter, Professor Wolfgang Ritter, who is the head of OIE Reference Laboratory in Freiburg, and he is also the president of the Scientific Committee Bee Health of Apimondia. Then we are missing Kurs Bielsberger. He might come later. Then we move to Aleš Gregoč from Slovenia, Agricultural Institute of Slovenia. And next to Aleš is sitting Jeff Pettis. Jeff Pettis is Vice President of the Scientific Commission of Bee Health, and he comes from the United States. Then we have Piotr Melzigi from Italy, if I read it correctly, Consiglio Nazionale per la Ricerca e la Sperimentazione di Agricoltura. Try. Fabio Sgogliastro, For Dipartimento di Scienze e Tecnologie Agroambientali, Università di Bologna, Italy. And we move to En Chen-Yang, who comes from Taipei, from Department of Entomology, National Taiwan University. And then I'm very pleased to have with us Peter Campbell from Syngenta, representing Syngenta, and Christian Moss, representing Bayer. Okay. So thank you all for being here. Thank you for willing to actively participate in this roundtable. And we will start from the end. So please, Christian, could you come to the floor? Simply start. So, would you like to move more close to me? Good.
Now it goes. Okay, good afternoon, ladies and gentlemen. Thanks for the introduction, and thanks to you all for being here.
For the translator, you must be
closer, yeah. So I'm pleased to be here with you today to discuss the topic of pesticides. And as you can imagine, we are particularly interested in particular pesticides. We are focusing a bit on the neonicotinoids today because that's a big topic of public discussion in these months. And I would like to give you the Bayer Crop Science position on in how far they are safe to bees and in how far restrictions of their use will help bee health. So, generally, neonicotinoids is a class of systemic insecticides which are used on a worldwide basis. Some are intrinsically bee-friendly and have a very low toxicity to bees. Others, in contrast, have a certain intrinsic toxicity potential. A particularly bee-friendly way to apply them is generally the seed treatment, because then they are around the seed and in the plant where they are needed, and the exposure of bees is minimized to exposure to very low doses. Nevertheless, there has in the last years been a considerable discussion about the safety of the neonicotinoids. And recently, as we all know, registrations of many products have been suspended in the European Union. A few words about how we are evaluating the potential risk of this kind of products and how the regulatory situation in Europe looked like. Of particular importance are here the so-called higher tier studies, that means semi-field and field studies, or studies which are conducted under realistic or semi-realistic conditions in the context of the whole bee colony under realistic exposure conditions. There's plenty of such higher tier studies available for all the respective compounds, and in none of those studies which have been conducted under realistic conditions there have been adverse effects on a bee colony level been found, neither by industry studies, and I'm also not aware of any external studies where a bee colony in its whole has been damaged by a realistic exposure scenario. There has likewise been a lot of discussion about specifically seed treatment as a new exposure route, the dust which can be emitted, especially when there is a seed treatment of a suboptimal quality. This dust is toxic to bees, of course, can for certain crops be emitted if not care is taken that the dust is staying there where it belongs. This potential problem has been recognized, and a lot of measures have been taken to cope with that, about harmonization of the required security measures, about optimization of the seed treatment quality to avoid the formation of dust, and with the drilling, with the sowing machinery to avoid emission of dust particles into the environment. So that we have now a situation where we have in the last years achieved a reduction of dust emission by several orders of magnitude. Another important piece of scientific information which leads us to the conclusion that those products are fairly safe under realistic conditions is monitoring data, as we have them from many European countries. This is for us the kind of highest tier evidence, because it's really observing the situation, the potential effects in situ, in the context of the agricultural practices, in the context of whole bee colonies. So monitoring are of key importance in their results. Monitoring data are available from plenty of European countries, and to our knowledge, in none of those monitoring projects a kind of systematic correlation between colony mortality and pesticides have been seen on a kind of large-scale basis. Likewise, I'm not aware of any correlations that have been found between overwintering mortality and exposure to certain typically treated crops. On the contrary, most monitoring projects really emphasize in their results the importance of pathogens and diseases as factor number one. To conclude, safety of neonicotinoids under practical conditions have been shown in a lot of studies which are employing a realistic design and confirmed by monitoring data. Therefore, we would not expect an improvement of bee health in Europe after the suspension of many products containing neonicotinoids. For sure, there are some open questions, some open points of scientific interest that should be addressed and can be addressed by appropriate scientific work. Just to highlight two examples, it is interesting to see how to explain this kind of perceived gap between laboratory data and field data. To give you an example, in many laboratory designs or otherwise artificial exposure designs, we see sublethal effects to individual bees, but we don't see those effects damaging whole bee colonies under realistic conditions. So it would be interesting to make a link and to explain that. So yeah, we are just involved in one of the studies going on this in terms of the FitBee project, to go after this in a multi-year study. Another important point would be to establish more monitoring data, and also here we are together with Syngenta participating or setting up a large-scale monitoring in 5 European countries to gain more data about bee friendliness, bee safety, or potential bee effects of neonicotinoids. Thanks for your attention.
Thank you. Thank you very much, especially because you were so short, just on time.
I try to be.
Yes. Okay. Peter, please. I forgot to mention that with us today it should be Dmitry Yanovich, but he was very busy, and Evgeny Rudenko will give us the pleasure to talk to us. Please.
Thank you, Fanny, and good afternoon, everybody. For those of you who don't know me, my name is Peter Campbell. I'm a scientist who's been working on environmental toxicology and risk assessment of pesticides for 22 years, and I currently lead the Syngenta bee research program. Okay? So, which button is it, Fanny?
Right.
Yeah, right one, thank you. So, let's go back one slide. So I decided to, because Christian gave you a very good introduction to the issue. Try not to repeat too much of what he said, but I'll answer the question in my first slide. And it is, I don't believe that the EU moratorium will improve honeybee health in Europe. And let me explain why with some of the evidence. Really, if you start to look at the evidence that's started to come out in the literature, you see that most of the evidence that has suggested there's a problem has been conducted in laboratory studies. If you look at the left-hand side of the slide, they're usually conducted either in the laboratory or bees are artificially exposed and then released into the field. And this is, if you look at most of the evidence that suggests there's a problem, it comes from studies conducted in that way. However, as Christian alluded to, on the right-hand side, there's quite a lot of evidence, certainly within industry, from the field, which looks at the real risk under environmentally relevant field conditions. Now this is to give you an example. This is a type of studies. I'm not going to read this slide, just to give you a feel for the number of field studies or semi-field studies that Syngenta has conducted for our neonicotinoid thiamethoxam. And you'll see that it covers a number of different crops, a number of different countries. It covers seed treatment uses, foliar uses, and all these trials have one thing in common. They showed no or very low risk to honeybee colonies under realistic field use conditions. And these trials are submitted to independent regulatory authorities for scientific scrutiny. And it's those trials that led to the original registrations of this product in Europe. However, I want to bring you to one particular study, and I want to show you some data. In 5 minutes is not much time, but I'll try and give you a feel for it. Yes, I'm getting too close to the mic, sorry. So I want to share with you trials that we did about in 2005 we started them, and it was looking at the risk to honeybees from the seed treatment use on oilseed rape and maize of thiamethoxam, and particularly focusing on the concern at the time was systemic residues in nectar and pollen from seed treatment use. Okay? And also to include in that study, because it had never been done before, to follow them for 4 consecutive years exposure and look at the overwintering success. Because most of the concern we've seen in Europe over the last years has been poor overwintering success of colonies that's led to major concerns for bee health. So this study was started in 2005. Bees were taken 4 years consecutively to the flowering maize or flowering oilseed rape and left there for the entire flowering period. And we looked at various parameters. You'll see there mortality, foraging activity, disease, overwintering success, and colony development. This is a quick slide to show you how we conducted the study. During the exposure phase of the trial, bees were confined right next to the flowering crop on the right-hand side for the whole flowering period, and then at the end of that period they were taken to a sort of semi-natural habitat where they could be excluded from other exposure to other pesticides, because the whole point is to try and understand, is there an impact from our pesticide? We took residues. So during that study we looked at the residues in pollen and nectar across both crops, and in general they were very low, between around the level of detection to 1 or 2 or 3 nanograms per gram. But we did get exposure, so that confirmed that the bees were actually exposed during our trial. This data is from, in particular, from our winter oilseed rape trial. Now I'll show you 3 slides with some data. This is a trial showing you the mortality data for honeybee hives at the winter oilseed rape trial. We chose this trial to show you because it was the one with the highest residues recorded in oilseed, in nectar and pollen during the trial. And you can see from this, the triangles are the control and the circles are the treated. And you can see there was, the control and treated colonies showed very similar forager mortality during the 4 years of the trial. Similarly, we looked at foraging activity, because sublethal effects, we worry about sublethal effects. We looked at foraging activity, and again, the same thing. The triangles are the control, and the circles are the treated. Very similar pattern of effect between control and treated, so again, no difference. And then finally, colony strength. We looked at colony strength, and again, if you look at those graphs, you cannot really see any difference between control and treated. The trends are very similar. So to conclude, from those long-term effects, where we took honeybee colonies and exposed them for 4 consecutive years, we saw no treatment-related effects of mortality, foraging behaviour, colony strength or colony weight, brood development, food storage or overwintering success. And we suggest that these trials better reflect the real or actual risk to bees under real field conditions from the use of our product thiamethoxam. And actually this data has been submitted for publication and accepted, and we hope to see it out in the press very soon. Thank you.
Thank you very much. I have to say though that you need to save your questions. Please write them down and you can ask them at the end so we don't break this order. Yes, please. And John?
my name is Chen Yang from Georgetown University. I would like to share the experience which we have done several experiments on the imidacloprid, this pesticide. As a neurobiologist of the insect, I like to work on the behavior and the physiology, and so this is a quite new field to me, but I got some surprise result. So okay, so we know that imidacloprid has been suspected to responsible for the mass death of the honeybee. In 2010, there's two papers surveyed do the review and then to see how the different insecticides, they are honeybee sensitive to the different insecticide or more sensitive to particular pesticide. But the result, it comes out that the conclusion is that the honeybee is not particularly sensitive to anything, any particular insecticide. But we know that in terms of the lethal dosage, that's nothing surprising. But we would like to see whether there's some lethal effect of particular neonicotinic insecticide. So we did a series of experiments in the laboratory. Okay. What I like to work is that it is a lethal effect, and we found that through a literature review we found that the LC50 is quite high, LD50 is also very high, but how about the sublethal dosage? And so we did, the first experiment we did is acute poisons, and then the second issue we would like to explore is about the chronic poison. And to see whether that's acute poison effect is, I did that's a simple experiment. You just need to have a feeder with sugar water inside the bottle, and let attract the honeybee to go to here, the forager go to here to suck the sugar water. Then you count, because there's a flow constancy, you can mark the honeybee, and then they fly between the beehive and the feeder. And you can count that after they leave the feeder, and you start your clock, and then you count when you come back. But normally, the honeybee, they come back in 5 minutes. So we did a statistic result here, and then you can see all the honeybee, they can come back in 5 minutes. But if you start to add some imidacloprid, then you can see that there's some delay, which means that the honeybee comes back later, more than 300 seconds. So we define that they are not normal. So there's a gray bar that shows that if you increase the concentration of the imidacloprid, then there's more and more abnormal honeybees behaviors comes up. And the purple bars shows that when the concentration goes higher than certain levels, then you can see that the honeybees they're missing. They may come back later, or may come back next day, or they never come back. Okay. This is also a result, and then we also look at these figures. We also count that some missing bees, they come back later, and then we start, because we mark them, and then we know which ones are which ones. So we start to count the intervals between they flying between the beehive and the feeder, and you can see it after several balls, then it becomes abnormal. Okay, this is also some result here. Okay, and then the second issue, I'd like to see that whether if the pesticide is so the sublethal effect is there, then it's possible that the honeybee that bring the contaminated honey or the pollen back to the beehive, and that can be the food for the larvae. And also we like to see whether this contamination affected the larvae. But unfortunately, that we count the capture brood rate and the pupation rate and the exclusion rate. Okay, this is the agenda which we work on this project, and unfortunately, that the result shows that it doesn't show that it affected the capture rate or the pupation rate or eclosion rate. So the student who did this experiment was quite disappointed because she expected that there's something there and may kill the larvae, but she was almost quiet for not being able to graduate to finish her thesis. But we saw that there must be something there because it damaged the nervous system. So I asked my student to start to work on the PER and to see whether those survive honeybee workers, so that when they grow up to be the foragers, and whether their learning memory ability is still normal or not. So this is the agenda which we use. We use PR. I think lots of people here may familiar with it. It's just a classical condition procedures. You can train the honeybees with the particular odorant, and then you offer a drop of sugar water, and then they can learn very very well, and then you give the odorant, and then they extend their proboscis. Okay, and so several trials you can train the honeybee, and this is a 2004 previous study has shown that the adult workers, if you feed them some imidacloprid, then you can see that their learning memory ability is just dropped very significantly. For us, we work on we feed the larvae with a very very tiny dose, as you can see on the figures, and then when they grow up, because we mark them and we know who is who, and then we mark them and we trace their behavior, and we know that even as tiny as the 0.04 nanogram, because we feed them 4 days during the larval stage, and for 4 days, and each day we feed them the 0.04 nanogram and or 0.1 nanogram. Then you can see that their learning ability just dropped very significantly. So if they grow up and then become the workers, then I suspect that they can't be very good for their future work as foragers. Okay, so that's my future work will continue to monitor those become stupid workers, whether they go out or they die in the beehive or whatever. Okay, and so, but the old techniques is not as good when you just look at the colony performance. So we started to use the bee counter to do that, and we modify the bee counters equipment with some wireless sensor network to report the beehive conditions every 30 minutes, and then this is what we did set up here. And then it comes out that the result comes very well, and we start to feed it in some imidacloprid, and then you can see that if it can become normal, and you added some insecticide, and then it dropped very quickly. Okay, but for some low dosages, they just dropped gradually. So we're still working on this kind of the pattern, the colony activities, in and out activities for the colony situation, whether they're affected by the very low dosage of the imidacloprid. Okay, thank you very much. This is all my presentation. Thank you.
Thank you very much indeed. Well, I would like to ask the rest of the speakers to be short, okay? Just to respect the time. I'm very happy to hear your results indeed, because I've been doing something similar, and but I will comment on them at the end. PEIs, the proposed extension, reflects the test that the honeybees is bringing her proposals out when she has a stimulus, okay? And it's a really very good test for testing this kind of substances. Please, Fabio.
Good afternoon, and so I want to say thanks, Fanny, for the invitation. It's a pleasure for me to be here. And so in this presentation, I will introduce the BEST. BEST is the acronym of the Bee Emergency Service Team in the framework of the Italian monitoring project BNet. So this new tool, this new service is want to study what's happening in realistic field condition when the bee dead. So we know that in the field it's very difficult to identify the role of pesticide in honeybee decline, because there are many factors in the field, because it's difficult to discriminate between poisoning and other causes of death, because there are limitations in the analytic detection of the pesticide in dead bees and other bee metrics. So for this reason, it's very important to have poisoning surveillance programs. So in Italy, since 2009, we have a national bee monitoring network together with the questionnaire survey. This is in the project Apimondia, and now we continue with this aspect with the project BNet. Together with this tool, we have the service of with direct inspection of the damaged hives soon after bee mortality. This service was implemented in the BNet with the BEST. BEST follows a standardized procedure when for the direct inspection of the hive. So the aim of BEST is to integrate the questionnaire and the National Bee Monitoring Network to study the events of bee mortality and colony loss when the causes are difficult to identify, and most important, to analyze the event in real time, when the phenomenon is still in action. Practically, every beekeepers in Italy can contact us, and we go to the damaged hive in order to identify the causes of bee mortality. And we follow a standardized methodologies. We systematically collect information, geographic and environmental data, the sanitary status of the apiary, and we sample several bee metrics for chemical and palynological analysis and pathological analysis. We take picture and video reports. All this data are integrated in order to understand the causes of bee mortality. So here we have a poster, so I invite you to see the poster, and where we summarize our results in 2012. So for each reports, we identify the factor involved. So we have sometimes cases of bee mortality where the main factor is the pesticide, other cases where there is the pathogen, or sometimes the interaction. Sometimes it was not possible to identify the factor. And so here we just to focus in when the factor involved are the pesticides. And we know that the bee mortality can link to the pesticide when the pesticides are correctly used. This means when the application is necessary, considering the economic and environmental cost, and according to the label and the gap indication. But we can have also bee mortality or colony losses when the pesticide application is done in an incorrect way. So when the application is useless and not permitted under certain circumstances, or even forbidden. So now we focus to these cases, and so I'm back here, Piotr Nedrisky. We'll continue the presentation showing some cases.
Thank you, Fabio.
Thank you.
Thank you, Fabio. Thank you, Mr. Lenin. And I will very briefly present you three cases of sorry.
What is this picture?
Kiev Metropolitan Station. And three cases when pesticides were mainly involved in the bee losses reported by our team. The first one is linked to poisoning due to illegal use of pesticides in citrus orchards in southern Italy during the blooming, and in several citrus orchards in Italy. Very often it happens that after the main blooming we have the secondary blooming with few flowers, but they are still there. So it is not allowed to treat these orchards. In this case, we went there to sample different matrices, and we have found a long list of pesticides, active ingredients, both in bee matrices and also in the citrus leaves. Of course, in this case, both neonicotinoids and other active ingredients were involved, and we have observed both high mortality rates in front of the hives and also other sublethal effects like abnormal behaviors and so on. We have received the reports about from about 150 colonies, but when we go to the place, we also try to estimate the total amount of hives involved, and in that case, it was about 20,000 of colonies. Unfortunately, among the pesticides we found in these matrices, there were also illegal ones, illegal active ingredients, and this means that growers use old stocks of pesticides, or even they search them abroad in other countries because they are convinced these pesticides are better than the registered ones. Totally in 2011, the ratio of illegal active ingredients found in our matrices was about 14% and 20% in 2012. The second case study regards the northern Italy, Emilia-Romagna region, and exactly Po Valley, which is a bit broader than that region. And I must say before that until 2008, so before the Italian suspension of seed dressing in maize, we received many reports of bee losses during maize sowing in the adjacent fields. So the last year before the suspension, 2008, we received 185 reports with bee losses regarding dead colonies. And after the suspension of the neonics and fipronil used for seed dressing of maize, in 2009 we have received only 2 reports about colony losses, and we went there to analyze samples, and in both cases neonics were used illegally, even if they were not allowed anymore. And totally, as you can see, in 2008, these 185 reports regarded more than 6,000 colonies. And the last case, the thing I want to add to the previous one is that in that cases, the pesticides until 2008 were used correctly and following the label. The last case I want to show you is a particular case also in southern Italy, particularly in Sicily, near Catania. And the orchards are also here citrus. And the treatment was done against scale insects, and there is a new species quite difficult to fight, to control. So the growers add some active ingredients to the mineral oil, and they are allowed. The problem is that the treatments normally are done not during flowering, but if the previous period is very dry, there's a lot of honeydew on the leaves, and after the treatment, this honeydew becomes contaminated. So in Italy, in Italian regulation, there's nothing about use of pesticides on honeydew. It's completely allowed. So in that case, the bees get stroken like after a treatment during flowering. In that particular case, we have received 3 reports in 2011 regarding 200 colonies, everything dead. But we have estimated, like in the previous cases, that all the colonies involved were about 4,000. And I know that this similar situation is in progress also this year. The period is more or less September. We will have the more fresh data afterwards. What I wanted to say with this presentation is that, of course, if the pesticides are used in an incorrect way, not following the label, it's very probable that we will have bee losses and bee damages. But in several cases, and these cases are not so rare, even if the label is followed, if the pesticide is used in the correct way, correct meaning following the label, in certain cases we may observe severe damages in bee colonies. Concluding, I would like to say that it's of course very important, and this is one of the outcomes of our bee team, that we must improve the communication, collaboration between beekeepers, growers, and veterinarians and bee scientists. All of them must communicate with each other, and also the farmers must be involved, also in training, in order to teach them how to correctly use the pesticides and to avoid damages for honeybees and also for wild bees. This is a part of our team after when we made it to register to this meeting. Thank you very much.
I would like to thank you, the Italian group. We invited a French group as well, but Luc Benjouses and Axel Decorti were really very busy to be here, so they are excused, of course. And I would like to ask Jeff Pettis. Please, Jeff.
Thank you. Good afternoon. I don't have a PowerPoint presentation, so I'd just like to make a few comments.
You will finish quick.
I will finish quick. Yes, I will finish quick. First of all, I have to start with a disclaimer, and actually this is a very serious disclaimer for me personally. I'm here as a private citizen because, as I understand it, my government has shut down. So I actually cannot represent the USDA today so I'm just here as Jeff Pettis from the US, but not representing the USDA. With that said, I'd like to make a few comments, and I look forward to the discussion that we'll have. That's what I'm looking forward to. So I've been involved in several studies where we've tried to look at field exposure. We've done survey work. The Mullen paper is often cited, where we looked at exposure to bees, across the country in the U.S. That was published in 2010, and we saw that bees are exposed to a wide variety of pesticides, not just one at a time. And then recently I published a paper in which we put bees on specific crops, cranberry, blueberry, things like that. And we see that there's multiple exposures at different levels. So we're never dealing with a single compound, of bees being exposed to, having that kind of exposure. And in the US, we run a lot of bees for pollination. So those bees go from crop to crop to crop. And so over the season, there's a cumulative effect. And I know that doesn't occur in many other countries, but for the US, it's a serious problem of multiple exposures over time. So I think I'd love to see more studies that take into account the realistic field exposure, and also more that chronic studies, and then put bees into wintering situations. And that actually may be unrealistic to ask of the registrants, but I think as a scientific community, we can do some of those studies. So we can make overwintering maybe the ultimate test for bees. The other difficulty I see is that we test bees on the individual level often, and then we try to extrapolate an effect at the individual bee level to the colony level, and that's very difficult. So, and even when we ask the registrants to do tests, most often it starts at the individual level, and it's really hard to extrapolate. So we need more, perhaps, tier 2 and tier 3 studies at field level. Lastly, again, similarly, we ask of the registrants to bring a product to market, and we ask that compound be tested, singly. But the reality in the world is that they're going to be exposed to that compound plus multiple compounds. And in all honesty, we can't ask the registrant to test all the combinations. So it falls to the scientific community to do some of that testing. Lastly, just to the harmonization effort, I think it would be great if we could come up, if we're going to ask companies to do more testing, more chronic testing, more sublethal testing, and longer tests, it would be advantageous to harmonize across the globe so that a product registration in Japan or the EU or the US, we're asking the same questions. Because I do think we need some more of these chronic tests, and they're costly and expensive. So again, I think harmonization would be a good thing across the testing. So that's what I have. Thank you.
I know that you have been doing quite a lot of work, so yes, I look forward to the discussion and then the questions of the people, and they can be answered by you. Alice, please.
Thank you, Fanny, for the invitation.
I hope you don't mind that we have allowed the speakers to speak only for 5 minutes, but presenting the results of the work has already been done through the health symposium session. So here it's only to give you a hint for the discussions, for you to have some input. Thank you.
Thank you. So in recent years, we made several tests on sublethal effects in honeybee larvae or adult larvae, and also we made some tests on colony level in the field. These experiments were made in 2008. So we can study some toxicological, physiological, behavioral effects, and also use cell biology and other methods to establish different effects on individual level. So as we see, these studies are not, probably easier to perform than tests in the field. So we also made tests in larvae adult bees, in adult bees also, and effects on tissues and also on colony levels. So I think that these tests are also very important, even those effects are sublethal. So stressors can induce, these stressors from environment can induce primary effect. This can be only detected on molecular level, for example, and secondary effect can induce some changes in DNA or cellular level, or can induce some respiratory changes. And tertiary effect can be demonstrated in colonies, on colony level, first time in cell structure, and then also in cell death or tissue damaging, and also on the colony level then in the end. So further, honeybee has also some mechanism how to respond on this to these effects of pesticide. So there is some buffering effects also on individual tissues and also on individual bees. We will not now go through this. So these tests are quite often performed on larval level, so it's possible to see changes in tissues, what is also useful. But sometimes we don't know exactly, for example, if there are some sublethal effects, if those effects are reversible or unreversible. So it's also important to know. So we performed different tests, morphological changes in tissues, detection of evaluation of cell death, so there is reversible or irreversible process in, for example, in midgut or in glands or so on, and then some tests on molecular level, let's say gene expression, which can be induced in larvae which are exposed to pesticides or also to pathogens. So it's important to predict physiological response to bees to chemical and biological threats. So there is also our aim to predict if we use something in the nature, also in the laboratory tests, what we can suspect from those challenging circumstances. So laboratory experiments which we carried on individual level in caged bees, and then also on larval level. So we made different tests on for example, in hypopharyngeal glands, in midgut, on ovaries, and so on. So on this level, we found effects of pesticides on individual bees. We also made tests about the effect of different pesticides when we performed tunnel tests in mice fields, for example. This was done in 2008. But at this level, at that time, we have not been able to show any damaging effect on, for example, on colony level. So this is, it seems difficult to demonstrate those effects in the field. Even if we find, for example, for the same substance, we can find changes in tissues and so on, but it seems difficult to find mortality or detrimental effect in the colonies. So it's possible to demonstrate adverse effects on tissue level in vivo. So there is also cellular response in organs and tissues. So there are also, in this way, there are also evident sublethal effects, which are also important, but we need to, as we also heard before, we need some connections, some further evidence about the effect on colony level. This would be very useful, even if we can find some intoxications and so on and so on. But it's not easy to perform experimental conditions to prove these situations. So field and semi-field experiences are also necessary. So this is my contribution, Fanny. Okay, thank you for your attention.
Thank you, Alex. Thank you for taking us from the field experiments to the laboratory ones. May I have understand?
Well, I will speak from here because I want to be short, and I can be short because we want to discuss these points. And I will only raise 2 points. One point is, this was mentioned that they have done, or may not, or you have seen, first I have to explain that on Monday we have seen a lot of different results on side effects of pesticides and neonics in our symposium of the Bee Health Commission, and I think it's not necessary to repeat them all. They have shown that it has been demonstrated very clearly that there are side effects on behavior, on pathogenity of some pathogens, and on life length of bees, and so on and so on. So I don't think it's necessary to go deeper into this point. But I want to raise one point, and this is the financial background of all this scientific work, you see. And if you go through the projects of side effects of pesticides, you find quite often different results and different conclusions of the scientists. And it's not easy for beekeepers or bee specialists and scientists to find out who is right. And not for no reason. Scientific research which is financed by the industry which is involved in this business are under under general suspicion. And so in my opinion, it's really necessary that in future we need more public funding on this research, and we need more independent results on this research, because I can say this as a scientist, you are never independent of those people who are financing your project. Even they declare that you are independent. You have co-workers in the background which have to be financed, and so on and so on. So I think we need more public funding on this research to get clear data in the future, and we should use the time which is given now by the EU, and hopefully it will be longer. And just to get results in the way that we can say, well, they are dangerous or not. So that's my statement.
Well, thank you very much. Saying that we need public funding. Okay. Gilles, would you like to add something?
Why not?
Would you like to stand here?
Hello, hello.
No. Could you please? No.
Yes. I am not a scientist, it's the first point. I just want to thank Jeff, because it was very short with his presentation. So he saved our time for the debate. So at the end, maybe we can collect some money for him, because he has probably... And I want also to thank him because he said one sentence about cocktails of pesticides. Is very important, of course, and it's nearly unrealistic to manage this. But when we are talking about field studies, in the fields we don't have just one molecule in our monoculture fields. So that is very important. Another thing, I didn't heard from the desk the word metabolites. That also is another issue, and it's very important. We must enlarge the debate with this kind. And I am sorry we don't have Etienne Bruno here, the president of the Technology and Quality Apimondia Commission. And just this morning he told me something new, because I was very busy. I cannot follow all the issues and in beekeeping. He told me that we have a new EFSA proposition coming mainly from the CARI work about the accreditation from the UN, from European Union, and submitted to SOFCA, in charge of this kind of problems. And he gave me some information, so I took some notes. And maybe if some people here more information, maybe in the debate you can add something. Just until now, for UN, it's quite simple, even incomplete. It's like a joke. We have labo tests with acute effects, DL 50, etc. And by contact and injection on adults only. And if we have problem, we go to the tunnel test, and if we have problems, we go to the field test. The proposition is first, about bee sensitivity, it's acute effect, DL50, but also chronic effects, DL50, in a period of 10 days. Also something to add, it's a larva test, and also hypopharyngeal glands development test. And about the bees exposed to the molecule, they want to add something about the spray, the high sprays and the low level sprays, and also include the dust, the problem with the dust. And also with the nectar starting at a level of 15% of concentration. We must include also pollen and also the contamination of water, puddles, and even guttation. Everything is included in the EFSA proposition. And also with a third chapter about wild bees and bambus, because maybe I am wrong, but tell me, the level of resilience is a little bit lower than our social bees, apis, etc. And of course, with this proposition, we can expect the phyto industry to be against, because with this kind of proposition, 90% of the molecules must be withdrawn from the fields. We have another problem to discuss. Is very, very important, because we are talking about bee mortality. I have just a very modest experience of more than 10 years of travels in the world. I visited and worked in 125 countries. I visited 45 universities, and until now we don't have the right databases about the mortality. We must work on this to be very, very serious on the beekeeping side. And of course, very interesting what you said. When you are in Japan or if you are in Argentina or in Europe, we have different, sometimes, criteria. It's the same molecules with the same bees, so we have to harmonize this. It's a minimum we have to do. Okay. But, Fanny, you told me that you wanted to introduce the debate now, but you did a good job also with your last paper. You have to say some words about it.
Anyway.
Thank you.
Thank you, Gilles. Thank you very much. I will wrap it up a little, and then I will talk about the agenda. Well, first of all, I'm really very happy, because I think that with all the talks that I tried to follow, we did address all the points that we had initially in our mind. Now, at the end, Gilles pointed out a few more, so we have more things to discuss about. Well, no, I didn't want to talk about my personal work, because there's some other work that we would like to talk. But it's really interesting what we hear till now. We hear contradictory results. And what I found in my research is that the bees, when you're doing the laboratory tests, bees really don't die. For 2.5 months, they just stay there, they are feeding. Of course, they are not doing any brood rearing, but they are there, they are doing whatever they want, but they are not dying. So you think that the LD50 finally, that it's absolutely necessary for the tests, it's crap. So what is happening though? Where are the effects? But they are there. Because if you see at the hypopharyngeal glands, well, because this I did, then you see that it's reduction. Of course, we don't know what's happening with the enzymatic activity of this, or what is the impact in the colony, but we have to look at it. Then if you look at the hearts, the cardiac system, then you see that it's going down and the bee is not the same instant that it should be. If you look at the respiration, you see that it changes the rhythm of the respiration, and the bee has asthma. If a bee has asthma and its heart is not functioning properly, how do we expect this insect to go out in the field and collect the honey we want to collect? Somebody asked me after my talk the other day, well, if you say that the bees are not productive, in China they have so many orchards, so many pesticides are going there, but they do produce still a lot of honey. I said, I didn't say that the bees are dying. Are not productive means that you have maybe depopulation colonies or whatever, and you try then, you feed, you cope with this, but it's costly. It's costly. And this is also only the laboratory experiments or the proboscis extension reflex that shows very, very good how the bees have a reduced ability to respond to sugar solution or to odors. I'm not very familiar with the experiment I did, very, very small one from the primitive stage of it, but it showed. It was there. I did not make it up of my mind. And then you go to the field experiments and you see that they consume the same amount of pollen, the same amount of nectar, and then when you leave them loose, you lose them. I opened the tunnels and I lost my foragers. I didn't know where they're gone, why they did not come back. Because while they were foraging in 12 meters, it was easy for them to orientate to their colony. And immediately I opened the tunnels, they were lost because they lost orientation ability. They could not find the way to come back. However, in the fields, from laboratory to semi-field and to the fields, it's a big, big differences. And this is another point that we should also look about how we can connect and have similar results from the laboratory to the field studies. Because yes, in the field there are so many other parameters. One year you have the population colonies. The next year, no problem at all. People are collecting honey in cotton. Why this is happening? But first of all, it has to do with the strength of the colony. A hungry colony will consume what you give it. A colony that is not so hungry will store it. But then you have the delayed effects that we haven't looked into yet. Also, there are multi-pesticides exposure or whatever. And there are also, I don't know, other kind of things, as well as different races, different tolerability, different. And till now, what we say is pesticides. Pesticides are the plant protection products. For me, particularly, if you want to listen to my opinion, pesticides are also the veterinary drugs that we use into the colony. However, today we talk only about neonics, maybe because this is the hot issue. But pesticides is also the veterinary drugs that we use into the colony, and it seems that they do have similar effects on our bees, and we have also to look into this. I'm only very sorry that the person from EFSA is not present, and Kurs Biesmeyer is not here to talk to us about the effect on the other bees, because it seems that, okay, yes, we are the honeybee group, but the other bees, they are maybe retaining or supporting the biodiversity in which we are based to make honey. So we should really think about it, and they are very, very sensitive insects. So I would like to leave it there, and thank you all for your inputs. And please, now it's your time. We are welcoming your questions or your positions. Yes, we do have microphones that they can go around. Yes.
We talked a lot about compounds of pesticides.
Yeah, microphone.
One moment, please. We have time, don't rush.
Thank you for everybody talking. We've had a lot of talks in...
Excuse me, Pierre, please present yourself.
Pierre Johannes from Canada. We've had a lot of information on the cocktail effect of all the pesticides. Jill mentioned metabolites. I would like to add something else on this list, and it's the adjuvants that we do not talk very much about. But let's say when we take the combination of adjuvants that are used to diminish the detoxification process in honeybees or in insects, but when added with the pesticide, the effect is multiplied by many, many more. And these adjuvants are present everywhere. And they are not controlled as severely as the pesticides. And I would like, if the panel could add something on this, because let's say like, I've done tests with bees and found piperonid octoxide in all of them. And as you know, this is an adjuvant that is used with pyrethroids. So I'd like, if anybody could add something, add information on this.
Okay, thank you. Could we have an answer to this? Yes, please.
In Europe, we have a different situation to the US. For example, before you register a pesticide, you have to test the formulation. So therefore, by default, you're testing the effect of the adjuvant and the active ingredient in that test. So I'm not sure if you do that in the US, but certainly in Europe we do. The other thing I'll say is, all the data, because we generate lots of formulation toxicity in Europe, when you look at that data, what you see is even in mixtures of active ingredients, the toxicity is driven by the most active ingredient. So we rarely ever see synergism. We know one case of it where pyrethroids, when mixed with EBC fungicides together, you can get a synergistic action, but that's well known, and pesticides are labeled to avoid that type of mixture. Okay.
One point to add, it's the same situation when we do a higher tier test. Then we also test the whole formulation, so the compound, the active compound, along with the adjuvants. So it's an important point, but in so far we can cover it quite well, at least with the testing system we have in Europe.
Thank you. Next question, or suggestion, or comment, or position? We are done.
Not yet.
Go home.
I had just a couple of comments. Introduce myself, I'm Neil Specht. I'm a custom pollinator in Canada. I make my living hauling about 4,000 colonies into hybrid canolas, worked with Pioneer Hybrid and Bear Cropside. So that's where I make my living. A couple of your comments. Franny, your cost of maintaining a hive is hitting the nail right on the head. And I agree with Jeff, the winter being the great equalizer. It tells the tale where we live. And we are seeing higher losses, some of them attributable to bad management on my behalf. But there are some strange things happening, and we're seeing, in my mind, I would like to see more investigation on the synergies with the surfactants and the fungicides that we're seeing used. Fungicides to me are one thing, but a surfactant, I understand that's like soap, it's just a water wetter. And if somebody poured a little soap on your steak, you'd just love it. Just an analogy. I know we shouldn't anthropomorphize.
Thank you. Any comment from the panel?
I believe we can see direct effects of fungicides in large doses applied 2 or 3 times, or at least twice in my case, with us standing in the field being drenched by it. Oh, it's all right, it's just a fungicide. There's problems with those bees coming out of that specific field that we know are, you can see the next year everything else has problems, but it becomes, it's something I'd like to know a lot more about. You guys are the scientists, I'm just saying, please look at that one too.
Professor Ritter would like to say something, and then Jeff.
I just want to add that, or just comment that we are very, it's very clear that the whole dilemma in which we are now, it's that everything is multifactorial, you see, and we have many factors, and of course bad management is one of the factors, and bad nutrition and so on and so on. So it's, we cannot talk, I think, today about all factors, because then we are losing time. I think we should talk about this one factor, pesticides or neonics.
Jeff?
Yes, I guess I just wanted to comment on the fungicide. That they are, by definition, often applied during bloom, which is when bees are visiting the crop. And I think the risk to fungicides has been underestimated. I think there's recent work coming out about that fungicides are having more effects than we earlier believed.
Yes, please.
In the discussion, if we can add something else, Pierre added the adjuvants. It's important also maybe to add the phenomenon of phytoremediation, how the plant can absorb and deliver the different substances. Phytoremediation, for example, is to, I give you just one example, to clean and to revitalize some industrial fields. We use some plants like pioneers in order to revitalize the soil. And in the world we use something like 6 different plants, and very interesting, one of the best one to absorb a maximum of poison in the soil is sunflower.
But if something...
I mean, when we are, if we want to connect this to our problem, I mean, if we are talking about field studies and molecules, we must know also which kind of culture. Maybe with the same product on a colza, canola, or on a sunflower, the result is not the same for the bees in terms of contamination of pollen, contamination of nectar, and even the droplets of mutation.
I would just like to add something about fungicides. Fungicides are also not good for bees, so they can cause quite great damage to different tissues. So once again on individual level, but very similar is also when we use acaricides in the colony. Even oxalic acid is not innocent, so everything can have some effect.
Yes, thank you, Alice. Well, I'm coming back to the phytoremediation. In fact, what we say now, we have 2 years of ban of neonics. And that means that for 2 years we are not going to plant any seeds that they are coated with these substances, whatever the substances, doesn't matter. However, if these substances are staying in the soil for 2 years, as some studies have shown, that means that they will already show up. And that also means that the previous years, or when we were planting these cultivations, we had additive effects. So I'm afraid that in these 2 years we will not see any difference or any better effects, a change towards better health of the bees, because they are still out there and they can just come up with the plants.
Fanny, can I respond to that?
Yes.
Because
1 moment, please. Yes.
In Syngenta, we've done crop rotation trials, so follow-on crops. We drilled maize after typical crop rotations, and we saw no increase in residues in pollen nectar. The half-life of thimethoxam in soil is 30 days. It's not years that I've seen in some of the papers that have been published. So there's a little bit, really these neonics are not accumulating in soil, and we're not getting higher residues in crops that are sown after a previously neonate-treated crop.
Okay, good to know, but we will see. I mean, if these 2 years we don't do anything, we don't do any studies, we don't take any measurements, we don't take any precautions, we should know what is happening in these 2 years in a measurable way, not only observation. Please.
Hi, it's loud here. Paul Kozak, provincial apers from Canada, from the province of Ontario. I did have a question, but I saw someone reaching for the mic, so it looked like they were going to be responding to Peter's comment. But yeah, I guess my comment would be related to what Fanny had said. Would be, you know, the persistence in the environment, and I have to, you know, disagree with Peter in that I have seen some data demonstrating that these compounds can persist in the environment. David Goulson has actually, yeah, and I'm not a toxicologist, so I mean Peter sure knows more about this than I do, but if I'm not mistaken, I think there is data to demonstrate that. In any case, if that is truly the case, should there not be multi-year testing, so to account for the accumulation of this in the environment? I think, Fanny, you had suggested looking at maybe higher levels that could build up in the pollen and nectar, in subsequent years. Pass the mic to you.
Yes.
If I can comment on your question, it's true that the degradation time of these compounds in soil may be variable according to environmental conditions. It's however important not only to consider the soil concentration, but also the availability of the residues by plants. That means the longer the residues are in the soil, the stronger they are bound to the soil matrix and become less available to the plant. And as Syngenta, we also have done kind of rotational crop trials where we had a plateau concentration in the soil, and we are sowing new treated seeds and likewise found that the residues in the rotational crops are either lower or at least not higher than with directly treated seeds.
Hi, Louisa Hubin, Oregon State University in the U.S. And I think you were actually making my point for me that there could be a lot of variation depending on soil type, weather conditions, all the different cultivational things that people do. But if that's so, then how can you just blanketly say that there's never that your experiment is the be all and the end all? You talk about possible variation.
I mean, I can take that. There's a good point, Goulson. You mentioned Dave Goulson's paper. All the data he presented were laboratory-based persistence data. And what we know about soils in the laboratory are quite sterile. When you test the persistence in the field, you always get faster degradation in the field, which is more relevant, okay? And it's a very good point you make. Persistence will vary from lab to field, from different soil types. The value I was quoting was specifically from our products in field soils in Europe. Around that you will get variability, you're right.
And of course we would not base such a statement on the individual studies. That's why we're doing sets of studies under different conditions with different rotation crop scenarios. Because we need to consider different types of crop rotation. That can be the fact under realistic conditions to have the overview. Okay, what kind of variability we have in the system? I fully agree.
I have to say I really appreciate how much effort you put into it, and it sounds like you really are focusing on it. And
Thank you for the comments. I do think, though, that we should continue with studies this particular 2 years because it's our chance to show the world or to us what is really happening. Yes, please. Olivier.
I'm Joseph Stieg from Austria. I'm beekeeper. It's possible maybe to lead this discussion on a more practical approach.
Yes, please.
It's very scientific. So the problem that we have, firstly, when always we get confronted with this issue, we have the problem, the industry, the plant growers say, your first problem is varroa. Okay, we have a problem with varroa. But if you go to a dairy farmer and tell him one cow has died off of a pest, you don't get permission to poison the next one. So they say, as long, so you can, in short term, as long you lose hives to varroa, shut up because of pesticides. So that's cynical. A dairy farmer wouldn't, and it's always the discussion is what's the damage? Is the damage just a dead hive or dead colony? And the percentage of the damage. So we had a discussion in our ministry. They said, oh, you didn't, the hives, they were not dead. Yeah. So it's like if I go out to the car and make a scratch with the key and say, okay, this car, it has the wheels, it has you can drive it, no damage on the engine. What you're crying about? So as a beekeeper, I have my bees. It's the honeybee. It's not just the bee. It's the honeybee. I have to produce honey. No one, even a hobbyist beekeeper, keeps his bees just because bees. But instead of that, he could keep bumblebees. So the honeybee is the honey is the key thing on the bee. So I cannot accept that someone says, oh, that's just 20% or so. So and even a dairy farmer wouldn't accept if you spray something in the air which would reduce his milk yield even 5%. So that's a little bit what I am pissed off, because always this discussion. We didn't choose the varroa. It's a pest, and we didn't choose it. We fight since 20 years with more and less effect against varroa. Okay, but that's not the permission to say shut up because there is varroa. So that's for me important.
Thank you. Yes.
I think that's a good point, and I
just only one thing I wanted to add. It was often mentioned the interactions between cocktails of molecules, but there are also interactions between molecules and pathogens.
Yes.
Serena, Nosema, and even varroa.
Just wanted to express that I fully agree that any kind of adverse effects, whether by pathogens or by pesticides, have to be investigated thoroughly, and that there should not be made a differentiation. This is important, this is not important. Yeah, both needs to be thoroughly investigated and treated accordingly, no doubt.
Yes.
Yes, I am Olivier Belval, I'm from France, I'm a beekeeper, professional beekeeper as well. And one comment first is that honey is our main production. Honey production in France has fallen down dramatically since the 90s. The 90s happens to be the time when a new type of molecules have been introduced into crops. This family, the neonicotinoids, are still present into the soil and are affecting our bees. Beekeepers have no interest in promoting one sort or another sort of plant protection product. Our main aim is try to get better off with our beehives. In the 90s in France, we were producing 36,000 tons of honey. Last year, 2012, we not achieved to produce 18,000 tons of honey in France, and this year it might be even worse. Honey production in a country is a good an indicator of how well the bee can survive in farming sector. Important differences are reported from beekeepers depending on whether your bees are in natural areas and even in city town centers. Sometimes bees are affected and don't succeed to produce honey, and in other times, in other areas, they produce large amounts of honey. In wild areas or in towns, in city towns, bees are not as affected as they are in agricultural landscape. These indicators, we've got them. Production, production is a very important factor for beekeepers. One point I want to make here is that European Commission is in the process of modifying its guidelines towards the analysis of pesticides. Why neonicotinoids are now sold in Europe and in the world? It's because the tests applied to these molecules were not adapted to the way of functioning of these molecules. Now we are in the process of having a partial ban of these products for 2 years for some crops, and these molecules should not be into the market. Now, the next step is how the new regulation will apply, and I would really like to know from the industry what they do feel about these new measures that have been proposed by the EFSA to the European Commission. Do the industry present here think this is workable for actual products, and would this work for the next generation of products? Because that's a real thing we want to avoid. We want to avoid to have a new generation of products that would appear functioning differently than all these or neonicotinoids, and that would affect the ability of bees to survive and the ability of honey producers to produce honey.
Well, thank you very much. I would like to comment before somebody else takes the step on the last two statements. First of all, I fully agree that because the beekeeper's aim is the honey production, that's why it's very important for any beekeeper, because most of us, we are beekeepers as well, to have colonies that they are productive. A colony that is not productive or is depopulated, it's alive, it's still there, it counts as a colony, but it's not productive. So this is a loss for the beekeeper. That's why we tried, or other people tried before me and before this group, to implement this kind of tests in EFSA recommendations. And it's really very good that finally, after so many years, that we keep on saying the authorities or EFSA organization or other organizations that they are responsible for the tests that they are going to be implemented for the authorization of a chemical, that LD50 only, it's not enough. And every time you need to take into account the mode action of the chemicals. And now we are talking, if we are talking about neonics, there are neurotoxic substances. So yes, of course, we should have gone into this direction years ago, but at least we did it now. Please.
Fanny,
yes?
May I comment to this point?
Yes.
If I may comment the last sentence, I agree completely with Fanny and with the other speakers about the productive point of view of beekeeping. Nevertheless, I think we shouldn't consider in the case that we still maintain high honey production with dying bees, we shouldn't consider the problem fixed, because we must remember about the environment. Remember that honeybee, dying honeybee, is indicating something wrong that is happening in the environment. So, productive point of view, okay, but we should remember about the other points. Thank you.
Yes, please.
I was asked a question, Fanny, directly by one of the members. The person asked for a comment from industry on the bee guidance. I think I should give him an answer, okay. Yeah? Because he asked the question. So, and also, the first part of his question is also relevant to me as well, because the product you referred to in France was our product, it was thimethoxam. Okay, so I can say a little thing. What I'll say to you is that the French regulatory authorities monitored the use of that product for 3 years, over 3 million hectares, looking at bee colonies and looking for effects, and they find nothing. Okay, so I can't comment on honey, because probably honey wasn't the thing they were looking at. They were looking at colonies and looking at the health of colonies, and they looked at 3 years of monitoring, 3 million hectares of treated maize with thimethoxam, and they couldn't find any adverse incidents or any effects on the health of those colonies. But going back to the specific question of the EFSA guidance, is a very good question. And I think Ms. Wolfgang, mentioned the EFSA guidance earlier. And the feedback I have from member states, the member state countries who have to use this within the regulatory framework, is they cannot use it. because they're asking for studies and where for some studies there are no methods, reliable methods that can give reliable results to address the questions that have been asked by EFSA. So the feedback we have from the member states is this EFSA guidance document can't be used in a regulatory framework.
But these new guidelines for the tests that they have to be used, there are methods behind the tests, they have been published methods. Maybe they are not real tests done, but for the methods, okay, yes. But this is the first step, this is the process that we have to reach. We should do that as well.
Yeah, I agree. I mean, there's definitely some improvements there, don't get me wrong. But for some of the studies, for example, for the field studies, the requirements for the field studies cannot be met. So you could enter a situation now where conducting a field study in Europe is impossible. And that's not good, because field studies give you a very good understanding of the risk under field conditions.
Well, that doesn't mean that we should not do them there yet. Okay? So, yes, Francesco Panella.
Just to answer
I'm so sorry.
The issue I'd like about the EFSA guidance. So about the field test. So in the previous guidance, the previous guideline of the EPO, the field study was really limited. So it was quite impossible to detect effects on bees, because you never know the real exposure for the colonies, and because the power of the test for the people, maybe many people don't know what this is, but the power of the test for scientists is very important in order to detect the effects. So the past EPO guideline was very limited from this point of view. So the new guidance document proposed by EFSA want to improve this limitation. So this was the aim of this EFSA guidance. So in order to improve these points, we have to increase the number of the colonies, and we have to increase the size of the treated field. We have to know the real exposure of bees. This is very, very important. We have to know the exposure of bees. Yeah.
Okay, please, Francesco.
Francesco Panella, European Beekeeper Coordination. Translate, please.
Okay, I was asked to translate from Italian to English.
Yes.
Come ai tempi del tabacco, siete sempre uguali.
Cosa vuol dire sempre del tabacco?
La discussione sul tabacco.
Okay. Like in the tobacco period, you are always the same.
Vendete un biocida. Vendete un biocida.
Okay, I think it's to you. You are selling a biocide.
E avete un atteggiamento ascientifico e ridicolo.
Oh, sorry.
No.
Okay. And you have anti-scientific and ridiculous approach.
L'os delle api è in un fenomeno di crisi, declino di tutti gli insetti, degli impollinatori e di tutta la fauna.
The honeybees are in a period of decline, not all, also regarding also other bees, wild bees and pollinators.
È la varroa che ha fatto perdere il 60% delle farfalle in Europa negli ultimi 20 anni?
Is it Varroa that caused the disappearance of 60% of butterflies in the last years?
È il nosema che porta alla sparizione dei bombi?
Is it Nosema that makes bumblebees disappear?
La scienza ci chiede di collocare un fenomeno nel suo contesto.
The science is asking us to find a framework of this phenomenon in a certain context.
È evidente che c'è un effetto multifattoriale, multipesticida.
Okay, it's evident that there is a multifactorial and caused by multiple exposure effect.
È evidente che la scienza, quella vera, non quella che voi pagate,
It's evident that the real scientific world, not the one that you are paying,
sta facendo dei grandi passi avanti per collocare questo fenomeno.
that this science is making big steps in order to put focus on this.
Io sono d'accordo con il professor Richter, la scienza deve essere indipendente.
I agree with Professor Ritter that the science must absolutely be independent.
Ma non sono d'accordo che sia pagata solo dal pubblico, deve essere pagata anche dall'interesse privato.
But I don't agree that it should be financed only by the public funds, but should also be there should be also the contribution, economical contribution from the private sector.
E in questo momento siete gli unici che traete utili dall'agricoltura mondiale e li dividete nei vostri dividendi.
And at the present, you are the only sector that takes advantage from the world's agriculture, and you divide them among you.
Basta vedere la chiusura nella crisi mondiale dei vostri bilanci. È incredibile. Siete gli unici che andate con le vele gonfie.
It's enough to have a look at your annual. Yes, President. You are the only ones who go.
Molto bene.
Okay. With the wind in on the back. Okay.
Business, very business. L'agro business, là. European Coordination Beekeeper chiede che siano implementate davvero e subito le linee guida scientifiche proposte dall'EFSA.
The European Beekeeping Coordination asks that the guidelines proposed by EFSA are immediately implemented.
Che sia rifiutato il ricatto che state cercando di fare.
Ricatto, aiuto.
Out. Refuse to out.
Okay, they request that the out that you are proposing must absolutely be refused.
Che siano al più presto possibile messi validati i test che EFSA propone.
And that the tests proposed by EFSA should be immediately validated. The last sentence that must be absolutely refused. Blackmail. You're blackmailed with the scientific word. I don't know this expression.
E propone che Apimondia si esprima a favore delle dell'implementazione delle linee guida dell'EFSA.
And the coordination proposes that Apimondia expresses his favorable opinion towards the implementation of the EFSA guidelines.
E che si consideri in queste linee guida anche un test fondamentale.
Anche un?
Test fondamentale. Un test fondamentale. Un test fondamentale.
Okay. And that considers itself a fundamental test in these guidelines.
Che parla molto, che è di campo, che è l'oming. Che è di campo, che è l'oming. Non senti?
Okay, and the test, okay, now I understand. The test, this fundamental test is the homing flight field test.
Io credo e auguro che l'atteggiamento dell'industria cambi presto per diventare un partner degli agricoltori e non il pirata che ci sta rubando la vita su questo pianeta. Grazie.
Okay. I feel and I really hope that the industry soon will become a partner in this effort and not an enemy that is stealing, pirate, that is destroying the life on Earth.
Thank you, Francesco. I would like. I would like the lady over there that she would like to talk for some time now, to have her comment, and then after the break, then we will have a break. After the break, you will have plenty of time to reply, please.
Okay, thank you very much. My name is Madeleine Chagnon from Canada. I'm a research scientist. I have been working a relationship pesticide in bees for at least 25 years now. This year, in 2013, I published a study, a paper with my colleagues on the effect of using acetylcholinesterase as a biomarker to show the effects of neonicotinoids on honeybees. We did our tests in the field, so this is really a field test. We've talked about a lot. Our field tests have been done. We have done field tests. We picked up foragers, and we had pollen traps. We know those foragers went into corn that was seeded with seed treatments, neonicotinoid seed treatments. We saw levels of acetylcholinesterase higher in those bees. And these sublethal effects should be investigated much more. Sublethal effects are very dangerous because bees are dying very slowly, and while they're dying, they're getting more and more sick. Neonicotinoids are cumulative and irreversible effects. So at the beginning, the bees will be working, bringing in the pollen that or the nectar that could have been contaminated. So sublethal effects should not be overlooked. And we did field trials, and we did find effects on the honeybees. We talked before, one of our presenters talked about useful use of pesticides. Neonicotinoids are insecticides. They were designed to kill insects. Honeybees are insects, so no one will get a Nobel Prize to prove that an insecticide will kill an insect. Okay? So it's for sure, and we have to use them safely and rationally, and seed coating is not a rational use to use pesticides or insecticides because they are always there should we need them or not. They are there, they are systemic, and they are everywhere, and there are too many molecules out there. All neonicotinoids have the same mode of action, so we test one product at a time, and many pesticides with the same mode of action are out there, and this is all accumulating and doing effects on bees. So I really approve the European position, and as my colleague here from Canada mentioned, we also have problems in Canada. We have persistence in soil, we talked about that also. Here in Canada it's cold, so we put everything in the refrigerator for 5 or 6 months. So it's for sure, we know that all biological processes are, when we put something in the fridge, everything stops. So the persistence is long because the product is not degrading during winter. So we get it the next year also. So I would just like to confirm the concerns that there are using neonicotinoids massively and more all over the world. Thank you.
Thank you very much. I would like to see your work. It seems that we have done several small studies also in Europe for field studies, but maybe they are not enough. And maybe in larger scale field studies would be better. Yes, please.
Well, a little bit the opinion that, or the feeling that you have the opinion that all laboratory tests are more or less garbage. And that's not true, you see, because you know that all pathogens have been tested first in laboratory, you see, and if you find any some effects between pathogens or cumulative effects, you have to go to the laboratory, you see. And you know that to repeat field tests is very difficult because the situation always changes, climatic influence, nutrition influence, and so on and so on. So I think we should not say, well, laboratory tests, and we do not observe them or we do not accept them.
Okay, no, we will have a break until 5 o'clock. Take your coffee, don't fall asleep, come back. I will leave you with one question. In my field studies, that, since 2009 that I started feeding imidacloprid, on my colonies it was the first time after the 12 years that I'm working in the institute that I saw American fallbrood in my colonies. How can you explain that? Okay, enjoy your coffee.
Just by chance.
Yes!
You're unlucky.
Bad management.
Yeah.
And please come back.
No.
We need you. Don't stay outside. Thank you very much all. Listen to me. Can we open it? Ladies and gentlemen. Nobody's listening. Thank you very much. If you could be a little bit more quiet, I would appreciate it. Before we start the discussion, I would like to call Evgeny Rudenko to talk to us. He's an Ukrainian guy.
Thank you very much, Mr. President, Mr. Chairman. We discussed very actual problems because it's not problem only of beekeeping. It's problem of all nations and all people that live on the world. Because we saw a very interesting presentation of President Bayer and the Syngenta company, but nevertheless, I'm not see a big foundation investigation of this research work that connected with using of pesticides. For example, in Ukraine, we have a law for implementation of using agricultural chemicals and pesticides. But some points of this law did not give the possibility to control some problems. For example, when pesticides registered in Ukraine, they made such instruction how we can use these pesticides. In your country the same. But it's interesting for me as a practical beekeeper and researcher how it's possible registered pesticides that include first group of toxicity for insects using by airplane. It's impossible. But nevertheless, all this company registered these substances and recommended using these pesticides by airplane. In widely days, it's covered a square that 3-5 times more than fields of some crops. Second one, you study now the problem of neonicotinoids. It's a problem, but it seems to me 12 years ago in Udine in Italy at Everbee it was first report that informed us that it's big dangerous for bees and especially for human health, for children health. Nevertheless, we recommended to use neonicotinoids and we recommended, for example, to isolate bees and make some chemical treatments using neonicotinoids, and isolated bees only 170 hours. It's recommendation of Bayer company. It's 7 days. It's impossible isolated bees in active period for 7 days. But nevertheless, 7 days, it's only period of acute toxicity. But next period, it's chronic toxicity. But never study this problem. I think that we must together initiate such proposition that this company must increase the fundamental examination of such groups of pesticides that very toxicity and very actual for our health, for our children, for our production and so on. Because I think that in different region, in different country, we have different contents of soil. And neonicotinoids, when they cover the seeds for growth, accumulated in this soil. And for example, neonicotinoids have, imidacloprid have period for destroying between 600, 900 days. It's more than 2 years. More than 2 years. If we study first period when these neonicotinoids increase the activity in first period, but we does not know what influence on pollen, on honey, nectar in second period. I think that we must increase research investigation, and this period that we receive when on 2 years in Europe was forbidden using of neonicotinoids. I write official letter to Bayer company and receive answer. Ukraine is not European Union country. That is why they claim to use neonicotinoids in future. I am not agree, because I want, if my child must be grows healthy, only. It's my country, and the lozenge of our conference, excuse me, funny, it's discover the European honeyland. It's our dialogue. I think that we must together help try to solve this problem. And I also thanks to Mr. President that he tried to discuss this question. Thank you very much, and I hope that we together make something step in this way. Thank you very much, colleagues.
Thank you also. So we promise that we will give the ground to the industry to reply to the comments of Francesco.
Okay, well, first of all, I'm going to start by saying I'm Peter Campbell. I'm not industry. I'm a scientist. Yeah, This is a very important point because this gets lost in this debate. So anyway, let me explain. First of all, as a scientist, anybody who works in environmental science as a career is passionate about the environment. I am as well. And I wouldn't do anything in my company that would make me conflicted in my beliefs about protecting the environment. So let's get that out of the way. Now I'll get to rational points. So industry, a lot of people have said, if the studies are conducted by industry, they cannot be trusted. Right? Those studies are submitted to regulatory scientists from the member state companies, all of the member state companies. They're submitted to EFSA, they're submitted to the Commission. They are not paid by industry, and they have to assess those data. So the fact that the industry fund those studies, I can understand how you could see there's a vested interest there, but there is no vested interest in the people that have to analyse the data. Okay? So all the data that we showed, that Christian showed and that I've showed, those studies are conducted to international guidelines, to good laboratory practice, and they're interrogated at length by member state authorities. So I'm quite confident that if there was anything wrong with this data, it would be picked up. Okay. It was also said that industry has no interest in beekeeping. Now, that's a very interesting point. Our industry is reliant on agriculture. Agriculture is reliant on bees and pollinators. It is not in our interest to harm bees, because if agriculture suffers, we suffer. So we have a vested interest in protecting bees, and that's a point that also is lost. Seed treatments, very interesting point about seed treatments. I actually think seed treatments are a great technology because it's targeted. It's targeted on the seed rather than spraying it from an aeroplane. And what's really interesting about this persistence debate we've had is that the people say, well, it's persistent, the residues can last. Our products, usually, by the time the crop is flowering, are no longer active against the pest. So that tells you they're not efficacious, they don't work against the pest at flowering. If they're applied, you have to then, usually to protect that crop, you have to have sprays later in the season. So if they're not active against the pest, that gives you an indication that the activity at flowering, which is the main risk phase for bees, is also going to be much reduced. Okay? And the last comment I want to make is, somebody came to me in the break and made a plea, and he said, industry versus beekeepers versus research, why can't we work together to fix the problem? I totally agree. I totally agree. I know Christian and I are both involved in collaborative research programs, working with researchers and with governments. We're doing monitoring studies on the next 2 years during the moratorium to try and generate some of this data. However, we have a problem, because as soon as we collaborate with a researcher, those researchers get attacked because they've been funded by industry. They get personally attacked because they're working with us. In addition, when we sign a contract with a researcher, we give them full permission to publish whatever they find. So we're not asking to hide data. Any contract from Syngenta, and I manage our research portfolio, they have full permission to publish their findings. So I'm again confident anything that we find in that research is open to publication. And that's why I believe we should be able to do this. I think working with governments, working with researchers to solve the bee health problem has got to be the way forward. And I wish we could find a way where we trust each other a bit more to do that. And that was all I was going to say.
Thank you very much. Relating with the red.
Hello, thank you. Thank you for microphone. I got the chance to participate to the EFSA meeting about the guidelines on pesticide issue. I don't know if you were there. So I can tell you that about 100 people were there. Among these 100 people, only 3 represent for beekeeping and beekeepers, and almost 80 for industry. We just got the explanation about the guideline and what the researcher or the researcher for the industry made. It was just complaining about each point. It's not possible to do that. It's too expensive. We cannot find this place in Europe. But what I can tell you that one researcher, an independent one from the university in Netherlands, I think, I don't remember where, sorry, did the experiment. So if one researcher can do this, you can do this. But it's not the problem of beekeeper if there is no enough place in Europe or if it's complicated for you. You produce this substance, you have to taste it. So the EFSA gave you guidelines, you have to deal with it, okay? And you just said before that you heard that the member states will not vote. The decisions are just right now and tomorrow. So how can you be sure that it will not pass? I don't know. So, and I have a question, more technical, if you agree. You said that you are doing some monitoring in 5 countries. Which one?
Okay, the 5 countries, coming to the last point first, is France, Germany, UK, Hungary and Poland.
Okay, thank you. And then you said that your experimentation about cruiser OSA just accepted for publication. Which one? Because in my university, for example, I have access to some publication, but not all. And sometimes you published also with some journals not allowed, who are not permitted, we don't have access. If we are studying something here or about bees, but not about chemical subjects. So that's why I'm asking which journal.
I can't tell you which journal because I'm forbidden by the journal. The publication will come out in a few weeks' time. It will be in an open access, peer-reviewed scientific journal, and I've been told by the journal I'm not allowed to say anything about this paper until the announcement from the journal is made. So I'm sorry I can't answer your question, but you will see it very soon.
Okay, next. Yes.
Yes, my name is Walter Hefricker from European Professional Beekeepers Association. I have a question for the gentleman from Syngenta. You were just praising the independence of government agencies like EFSA. Could you explain to us why your company threatened the director of EFSA with personal liability in the publication of the neonicotinoid results? The legal documents that were written by Syngenta's attorneys were leaked and are open to the public, so anybody can read this. Second, after EFSA reached its conclusions with your input, not just EFSA staff, but also the risk managers of the member states. And the Commission then turned this into a proposal. This proposal was adopted in the case of the 3 neonicotinoids in the second round. In the case of fipronil, after member states had could think about their position a little bit longer, it was actually adopted in the first round, which is highly unusual for any kind of environmental measure. Could you explain to us why your company and Bayer are now challenging this decision in court? Thank you very much.
With pleasure. I mean, I mentioned that the EFSA and the member state authorities are independent. Yes, they are. It doesn't mean we always agree with them. Yeah. So in the case, EFSA, we openly, Bayer and Syngenta are taking the European Commission to court because we believe they have not followed. They've changed the rules of the regulatory process. And also, when you look at their assessment of the data, they ignored many of the studies that we showed you today were not taken into account at all. So we're not comfortable with how EFSA have reviewed that data package. So that's just a difference in scientific opinion. Okay. I can't comment on the letter from Syngenta because I didn't write it. I have no idea what it said or even if it exists. But clearly, on the neonics, we firmly believe our products are safe. We've got years of use in many countries where with monitoring in place and saw no effects. I've seen restrictions in place in France in the 90s when neonics were forbidden for a while. I've seen restrictions in Italy, where they've been forbidden. And I've not seen honeybee health improve. I still see overwintering colony losses around 30%. So, you know, I'm not convinced as a scientist that the pesticides or neonics are the problem for the honeybee losses that we see in Europe. And that's my scientific opinion.
Yeah, maybe to add some words from our perspective, yeah, we of course fundamentally agree. It's the process that we are challenging, that for instance EFSA evaluated seed treatment and granular treatment, but the Commission proposal also covered spray treatments, so something that was not even evaluated by EFSA. And the other thing is, or one of the other things is this kind of retrospectively applying a new legislation. This is the point we are challenging.
Okay, thank you. Maybe it's not good to have a discussion, maybe somebody else, but I would like to give the ground to Piotr first, because Piotr? And then, yes, please, we will do your questions. For the...
Yes. I have one preliminary point. I must say that I'm very sorry to contradict you, but time before my institute carried out a study in collaboration with an industry. It was only once. We did it only once. It was 2002 and 2003-4. And the topic was a study on colony health in maize sowing period. 9 sites were involved, and we got financial support from the industry. And we weren't allowed to publish the data. And we still have all the results in my office, and I'm not allowed to publish it. Even if before it was said that of course we will publish together. Anyway, the other side is now changed its mind. Okay, the thing I wanted to say, and that's why I asked Fanny to give me 2 minutes, is the explanation about the discrepancy between the results, most of all field results obtained following the same procedure, but conducted by different persons. For example, industry and universities, or even different universities. Let's start from the point that in case of neonics, for example, the literature reports very huge spectrum of LD50s. I can carry out a laboratory test simply on bee toxicity, bee mortality, simple lab tox test, and I will obtain one value, you will obtain another value, even if we follow exactly the same procedure. This means that in case of this particular group of pesticides, there is some factor that normally is not considered in our experimental design that has strong influence to the result. And that's why the same procedure may produce different results. And for the same reason, you may, in the same study, we can obtain different results in field studies. This will be even more likely because in the field, of course, we have much more external factors that are not considered in the experimental design. And for the same reason, and I am finishing, the study carried out in one site by one institute may not show statistical importance of a treatment. For example, for the reason that 10 units of untreated group have very low, let's hypothesize, mortality results. Among them, 9 are completely healthy, and 1 is ill. Let's say, let's use this term. But in the treated group, we will have, for example, 5 dead, 3 ill, 2 healthy. For the reason that I said before, there are many factors that influence the result, the mortality results in case of these compounds. And in this case, even if it's obvious that the control group has reached much higher mortality rate. The statistics will not confirm this obvious results, and this is one of the problems. The commonly used statistics very often don't show such an obvious difference. And this is linked to what Fabio said in his presentation, the power of the test. And this is one of the reasons why the new EFSA guidelines has improved the field trials in order to enforce the power of the test. You cannot obtain statistical difference using 4 colonies per treatment. You will not obtain, you will not see the statistical difference, the effect of the treatment, not because there is no effect, just because there is no statistical difference caused by the high variability of the treated colonies. Thank you.
Thank you, Piotr. Maybe this is why we need to have large-scale experiments. But the thing is, think only one parameter, how far the colonies are from the crop that we are testing. They might be quite far, they might be close. Yes,
hello.
The gentleman over there.
Hi, Mike Alsop from South Africa. I've got 2 questions. The first one is about the harmonization of standards and procedures, which I think everyone would think would be a very good idea. From the panel, any idea, well, sort of a twofold question. How realistic is that, given the different environmental conditions and the different bees at play, and also the different priorities of the different countries? Some countries more agricultural and other countries more hobby-related. So how realistic is that harmonization, and any idea about what practical steps could be taken to move forward to try and achieve that goal? And the second question, I hear about, I don't know the details, if it's Bayer or Syngenta or combination, monitoring 5 countries over the next 2 years. I'm not quite sure what you're monitoring. My question would be, over the countries, the crops that the neonics are currently or have been used on, they're still going to be planted over the next 2 years. Growers are going to use old generation organophosphates and pyrethroids on them. Surely the European Union or EFSA or someone should be monitoring the impact of those old generation pesticides in comparison to what has happened in the last few years, rather than relying on the chemical companies to be doing the monitoring. Thank you.
Somebody would like to answer?
I mean, yes, definitely. About the point of the harmonization of testing requirements, I mean, this is something that we definitely would welcome, to have kind of consistent requirements, not only in one country or in one region. That's a good thing. If there are reasonable requirements, we are absolutely willing to consider them, and the harmonization can only be beneficial. The point about what is coming instead of neonicotinoids, what will be the replacement? Well, I can just support the question, because it's an important question. We really need to ask ourselves, will the replacement be more beneficial, more soft to bees? I just leave this question open.
Thank you. However, I put down here a question says alternatives, because I would like to ask this myself.
5 seconds.
Sorry?
5 seconds. I'm asking 5 seconds.
Yes. But do you have alternative suggestions for the people that they don't want to use neonics?
Well, of course there... First your 5 seconds? All right. Of course there is a broad spectrum of different products that can be used in different cropping scenarios. It's mostly not the case that one is 100% replacing the other one or compensating the other one. Most are more or less tailor-made to the pest spectrum, to the crop, and so on. And there's also a certain level of diversity relevant to avoid the buildup of resistances. That's one point. About new alternatives, about new compounds, I mean, of course we are regularly developing new compounds, new actives, and of course bee safety or bee friendliness is for us a very important parameter already to check in the early stage. And we definitely look for compounds which are intrinsically bee friendly. Of course, we have to consider a lot of other factors. I mean, they have to be non-toxic to humans, they have not to leach the groundwater, no endocrine effects, and on the other hand, they also have to be efficient against the pests. So it will always be a certain balance in there.
Okay, thank you. Piotr? 5 seconds.
Yeah, I completely agree with my colleague. And but I would like to add another possibility to substitute the neonics, just to not use anything in certain cases. In many cases, these treatments are simply useless. For example, seed dressing is a treatment that very often produces no effects. In terms that we have the suspension since 2008 in Italy, and there are fields where no damages are observed in maize, in corn, since that time, even if nothing is used to protect the seed and the plantlets.
Yes,
Thank you.
That's fine. The thing is that the gentleman over there asked also how we could explain or how we can see that there can be harmonization. Can we have an answer to that?
Sorry, if I can just narrow it down. My question on the second part is specific. Is if you withdraw something, then you surely must evaluate it. Who is evaluating it? Who has said, so now they've said across Europe, you're not allowed to use this. Someone must surely be saying we are going to assess whether it's worked or whether it hasn't worked. Who is evaluating it?
I'll answer the question very simply.
Or is it just politics?
Yeah, you're right. I mean, the problem is, to my knowledge, there is no direct commission program to look at that. There are some programs at member state level, so I'm aware that some of the member states, so not industry, but funded by the government institutes, are trying to do some monitoring in some individual countries. But there's no concerted effort to look at that at all.
Thank you. But I don't know if you were here before, but we said that these 2 years that the specific neonics have been bound, we need to do something. So we need to find ways, methods, I don't know, funding, to do research and find out what is happening. So we will monitor. I don't know how, we have to. Jeff.
I just want to reiterate what Piotr said, that I think we've moved away in much of agriculture towards just almost preventative treatment and not towards a more integrated approach. When the pest is present, you use the product. I will say there's less exposure with seed treatment, but I think we're overusing them as just prophylactic, not when they're actually needed.
Next.
And there are also other tools, agronomic ones, for example, crop rotation. Okay. Which is very useful to protect.
It's also a kind of money which is more expensive, which is more cheaper.
Good farming practice is replaced, but the alternative is replaced
farming practice.
Microphone bad.
Yes, Maribia. I've seen a couple of other people, Norman.
Hi, I'm Maria Joseph from ANSES and the EU Reflab for Honeybee Health. I have a cheap question, first of all, for the two gentlemen from the scientific gentlemen from the industry, and also for all these scientific gentlemen from the public sector. We all know that experiments have demonstrated that pesticides have effect on honeybees in the lab experiment at very low doses. We had a few examples on the presentation, but there are many experiments published on that. We also all know that the colony have a buffer capacity to absorb some losses, and they have a very complicated system to compensate these losses. And this is why we cannot observe this very fine effect in the field. So my question is, what would you propose as technical issue or protocol to study this buffer effect in the field, please?
Could we have the answer first before we go?
Sure. I mean, I think you're right. The problem is the honeybee colony is a very complex organism. We call it a superorganism, maybe because all the individuals working together with differentiated roles. We've got a project in Syngenta which is looking at trying to model the dynamics within the colony, so trying to understand, to develop models that can predict how colonies will behave under different stress situations, not just on pesticides actually. They're looking at forage, how they respond to forage stress, how they respond to temperature. And so we're starting to build a model. It's called Behave, but it's been developed with some academics in Germany, and it started to look quite interesting. But it's very complicated because at the end of the day, it's a model, and you have to test that model with the field, and that can be quite challenging.
And
well, yeah, I think that's a very important point. As we have seen, there are a lot of data from the laboratory that suggests certain kind of effects which we then don't find in the field. I must admit I don't have the silver bullet design that could kind of bridge this gap. As Peter pointed out, I think modeling could be an option. It would of course need to be validated, would be a kind of long-term thing, because the key thing of modeling for such a complex system like a bee colony is validation. That could be a direction, but there may also be other directions. So I think this is still a field of research where I would really see a lot of very valuable studies could still be done.
Any other answer?
I think that large-scale experiments can contribute to discover this, or to help to open this buffering effect or ability of colonies. So large-scale experiments which are under some kind of scientific control.
Okay. Yes. You, yeah. And then Norman.
Thank you, first. Pam Hunter, UK. I'm a small-scale amateur beekeeper, but my background is the pharmaceutical industry. So I spent all my working life working on testing drugs for humans. And when I first started to take an interest in this field, I found it very difficult because it's actually easier testing drugs for humans. It's done at a laboratory scale. I don't understand why people criticise these tests for the laboratory. All the drugs we test were developed in a laboratory for us. And you start off after the LD50, you have 7-day toxicity, 14-day, 1 month, 3 months, 6 months. Rats and dogs, usually rats and dogs. And you're looking for target organ toxicity. And that effectively is what an awful lot of people have been doing. They've looked at effects on the hypopharyngeal gland, all sorts of things. And a drug gets passed quite frequently at a level which is thought not to have too much effect, or a multiple below it. And when it's been out in the clinic, quite often, once it's been dosed, there's been a couple of examples, it's not till it's gone to about 3 million people that the problems start to show, and that drug may be withdrawn. But going back to these laboratory tests, a very low level, let us say kidney effect, it will take a long, long time of you taking that drug before it shows up. Remember smoking? How long did it take anybody to accept that? And when I went into the industry, we were all horrified by thalidomide and DDT. And the reason those drugs got through was because people didn't understand sufficient. And I really do just make a plea to say, don't ignore all these laboratory tests. I do appreciate, and I would just like to thank publicly these two being brave enough to come here. I do think we should congratulate them, but please, the bio gentleman, Dr. Mouse, please don't call them bee-friendly. Call them the least toxic. No insecticide is bee-friendly. And I do worry about this gulf. There is this gulf. I respect what you've been doing, but I do think we have also got to respect these laboratory tests. These compounds are doing things, and I know the dilemma is at what level, but I just keep thinking of the human side.
1, 2, 3, 4. So goes to Norman.
One comment. I mean, it's absolutely right. I don't say that laboratory trials have no value. They are important. They can bring very important insight into how a compound works and so on, how the intrinsic toxicity is. The thing isn't only that in the case of the bees, frequently certain effects that we see in the laboratory, we don't find again in the field. And that's what I meant with there is still a gap that we don't understand. And then, well, right, I mean, that's, you came with the example of smoking, that's a very good one, I think. I think here it should be possible to find under field condition a correlation between certain kinds of cancer and exposure. And that's the thing, in the projects, in the monitoring data I'm aware of, we don't see a correlation between exposure and colony mortality. So I think there is still a gap we need to fill. I fully agree with this.
Yeah, thank you. Professor Ritter would like to say something.
Well, I just want to mention that I totally agree with you. And I think we are absolutely in the same situation. So everybody who is doing field tests knows, and I repeat what I said before, that not every year is the same, and we have always difficulties to repeat some things. And if we see something in the laboratory, maybe we see it 5-6 years later in the field when the situation is critical enough that these and all the factors come together, that these factors become a big influence. So again, I totally agree with you, and we should recognize these laboratory tests. And if we start with field tests, they should be on a large scale and for a long time.
But if I remember well, in the last discussion we had many of us in EFSA, that was one question, how we can transfer or repeat the laboratory experiments in a similar way outside in the field. And then maybe something for the gentleman from Africa. We're talking about harmonization of methods. First of all, we need to do the large-scale studies in different areas, in different environments, under different genetic material to know how this reacts, to have the basis and find out the solutions later on. Norman, yours.
Just on that final point, which wasn't the one I was going to make, one step in the direction of harmonisation, I must just plug the bee book yet again and the chapter on toxicology that Pietro has put together. It's imperfect, it must be, but hopefully it's a start, and we must all contribute to improving these things in the future so that we do actually do the same tests under comparable conditions. But I was actually originally going to make the point that Mike Alsop made second about what's actually going to happen during the 2 years of this moratorium if we don't use neonicotinoids. And my background actually is I was an agronomist before I became a bee scientist, and therefore I've been interested in growing and protecting crops. And I have colleagues and friends who are also involved in protecting crops, and I've asked them recently what their clients will be doing next year if they can't use the neonicotinoids. And I should say, from a UK point of view, the crop that is most relevant to this debate is oilseed rape, which has almost entirely been treated with neonicotinoids for the last 10 years or so, and the chemicals used have changed, and imidacloprid, that we've heard a huge amount about, actually is hardly used in the UK on oilseed rape anymore. I think if you asked most of the farmers about integrated pest management, they would actually say that they are using these seed dressings as part of an integrated pest management policy. And the reason they do that is because they know that if they do not treat the crop, they will have problems with cabbage stem flea beetle and pests that attack the crop in the autumn. And in the UK, the particular problem is that the soil conditions are frequently unsuitable for actually getting on that crop to spray with something if you wait until you have a problem. And they have found these things to be incredibly successful and useful. So we're left with the question of what they're going to do if they can't use neonicotinoids. They could, of course, stop growing oilseed rape completely, and you have to remember that oilseed rape is the major honey producing plant in the UK, and if farmers stopped growing oilseed rape, that would greatly affect the forage available to bees. But that's not going to happen, because if you look at the gross margins of crops that are grown, oilseed rape is up there, sort of number 3 in the list behind sort of feed wheat, milling wheat and so on. And it fits perfectly in rotation, sort of 1 year in 5, and it's an extremely useful crop. So they will go on growing it, and the question then is how they will protect it. And most of them don't know quite what they're going to be doing, but they may well be using Durspan, which is an organophosphate, which I certainly would not suggest is something to be recommended, or they will be using some of the synthetic pyrethroids. And I think the problem in this debate is that we're moving the goalposts all the time, and we're demanding, quite rightly, ever-increasing improvements in safety. And in the same way that we're talking about subtle sublethal effects of neonicotinoids now, and that the problem is we don't know enough about those subtle sublethal effects, equally, we don't know very much about the subtle sublethal effects that these synthetic pyrethroids may have. And we can look at the actual experience where these sprays were used in vast quantities for many, many years, and we can look at the colony level and say, oh, they're absolutely safe because they weren't wiping out colonies, whereas 10 years earlier, when organophosphates were being used widely, lots of colonies were being wiped out. But that doesn't mean they have no effect on honeybees, and you know, we simply don't know enough about these older compounds in particular. Part of the reason why there is little demonstration that they were harmful to honeybees is that the advice to farmers was to avoid spraying in the middle of the day when honeybees are foraging, but spray early in the evening, early in the morning and late in the evening, which of course is precisely the times when bumblebees might be foraging, and we really don't know what effect those were having. And I think it should be something that we bear in mind, that before you make a major change in anything, you should do a risk assessment as to what the implications of that change may be. And I'm not convinced that anyone did any risk assessment before deciding this moratorium. So, just to finish, we must use those 2 years wisely, because we don't want to be having exactly the same debate in 2 years' time that we are having now. We must go out and do those studies. And we have the problem that we have all these laboratory studies that show that things happen, and we have a range of field studies. There have actually been quite a lot of quite big field studies that don't tend to show statistically significant results. And it could be that that means that there are no effects in the field, or it could be that we've done the wrong experiments. And it could be that we haven't done experiments that are big enough to have the statistical resolution to actually show those effects. And from that point of view, I'd like to reiterate what Wolfgang Ritter has said, that governments need to put some money in the next 2 years into carrying out those very large-scale field experiments that hopefully would show those effects. But equally, I hate to use the word industry, I've talked to lots of your colleagues over the years, and they tend to say, oh yes, we did those field studies 10 years ago, we've got all the data and it doesn't show a problem. It's great that Syngenta is publishing this one paper, but you have huge amounts of data that the scientific community really would like to see and really would like to evaluate, and I would like to make a plea for more openness.
Thank you.
A few items here. This is Paul Kozak from the Ontario Ministry of Agriculture. One thing I think we all could do is to communicate a lot better with each other as far as from different regions, different countries. I say this with the 2 years of a large number of pesticide incidents occurring in Ontario, and this was specific to corn seed and the seeding process, at least what we understand so far. And this has been happening years earlier in Quebec, and we weren't really aware of that. I wasn't really aware or didn't really appreciate what had been going on in Italy, and now I'm really coming to appreciate all the work that that's been done through AidNet and BNet. And again, then we've got these incidents that are now happening in the Midwest of the United States that Christian Krupke has been working on and highlighting. The other thing I think we could do is, I think we have to be, we really need to define things better. So when we talk about honeybee loss, a lot of the focus is kind of lost. In Canada, we do focus mainly on winter loss, and we haven't really defined mid-season loss as much. And I think, you know, to talk about honeybee decline, honeybee mortality, we really have to define that much better. I don't think we've done that as regulators and as research as well as we need to communicate to the general public. The other thing I think we have to be specific about is different crops, different regions. So we've got all these, you know, we're all here to talk about neonicotinoids and bees, but we have to keep it specific to the crop and look at some of these broader properties of the compound and fit it into that. And what we've been dealing with specifically has been the corn incidents in Ontario. The other thing is that monitoring keeps on coming up, and I think that is a really important part. I can say that with all the work that Quebec has done and Ontario has done, we're still not where we need to be. I do believe that this should be something that should either come from research or from government. And main key in that is the whole concept of conflict of interest. It's something that we deal with all the time in government, and it's taken very seriously. And conflict of interest is not just, it's not just the reality of it, it's also the perception. So, you know, there may be a lot of people in this room right now that, you know, when they see something come out from industry or, you know, a scientist that works with industry, it really does undermine the credibility, whether there's a reality to it or not. So I think that's the secondary reason that, you know, we really do need a third-party and objective source of funding. This monitoring really is important. We're asking some very tough questions right now about what is going in the water, what's staying in the soil, what's being expressed in the plant tissue. And we really don't have those answers right now. We don't know what to tell our beekeepers from government with that. And that's largely because the data has not been collected on that. The other thing about harmonization, I think there's going to be some realities with harmonization in that, you know, we've got the European Food Safety Authority with their decision, we've got the Environmental Protection Agency through the United States, and we've got Health Canada through Canada. And at various different points, there may be very different opinions on the data. Right now, I mean, the whole concept of a seed treatment being much more safer for honeybees. I think it was 3 weeks ago that Health Canada had said that the current use of corn and soy seed treatment is not sustainable for pollinators in Canada. That's a very serious thing for them to say. I think it's largely been driven by incidents that are being reported to the government, that are being validated through residue analysis. But I'd just like to highlight that one jurisdiction or one country may have a very different take on things. So I think that's going to be a challenge for the harmonization. I was going to make another point, but I think someone made it for me that, I'll just continue on it. Field relevant, just speaking about the incidents that we've seen in Ontario, and this is not a research perspective, but it's coming out into an incident and responding to it in the field as it happens. What the beekeepers reported, going there with the pesticide agency and the bee inspectors. And we found that in some cases, you can have 28 colonies in a yard, and only 2 of them may have gone to where that pesticide was. It all goes down to the biology of the bee, where the scouts are going. They could have just picked the wrong side of the field. And so I think that's going to be a real challenge for field relevant studies and how that needs to be addressed. And, you know, it is a really good question about what people will use instead. But we do have researchers that are looking into that. We've got researchers in Quebec from, I think, the Department of Agriculture. It was someone that presented in one of your meetings. And Christian Kruppe is looking at, are these actually resulting in an increase in yield? And I think that has to come back to prophylactic use of a treatment, and what even some of these crop specialists, pesticide specialists, are challenging as true IPM, if you're just putting something on a seed, whether you need it or not. And I'd just like to end with a technical question to Peter Campbell. What was the acreage in that study? Or
which study?
The study that you had presented.
Okay, cool.
How many acres was
Yeah,
it?
You don't think of it in terms of acres, you do it in plots. So it's a replicated field study, plots of 2 hectares in size, and then they're separated from each other and controlled by about 2 to 3 kilometers to try and get isolation. So you're doing it in 2 hectare plots, which is enough per plot to provide the bees what they require.
Okay, so great. Okay, that's great, because I mean you're doing field relevant research then, right? So what area would 2 hectares be field realistic for crop use for canola?
This is a tricky one. Can I turn the question round a little?
Sure.
Because it's come back to the statistical point made by our colleague here, which is a very good point. To get statistical power, you need a high number of replicates. So it's a number of replicates that gives you statistical power. And in this case, the honeybee hive is not a replicate. If you put 6 or even 20 colonies on the same plot, that's not a replicate. It's not, because they're not independent from each other. So you have to replicate to the plots. So EFSA did an evaluation recently, which has been mentioned in the guidance document, where they say you need 14 pairs of 2 hectare plots separated from each other for control and from alternative forage that used to forage by 2 to 3 kilometers. When you try finding that many, to get that level of control, replication and separation in an agricultural environment is virtually impossible. I understand why they're recommending it, because giving them power that my colleague here wants. But to actually conduct a field study in an agricultural environment with those sort of requirements is impossible.
So you're saying that the current agriculture...
Excuse me. I think that we wouldn't like to go into very technical discussion
Sure, yeah.
of how we can plan or how we can do experiments, because this is not the point of this conversation. We've gone too far now that to go back to very many details. I think that the most important is something that we didn't. I would suggest that we have already 2 questions, 3. Yes, 3, because I saw it before, and we will stop it there and then conclude and see what we can come up to. But before that, I didn't hear from any of the beekeepers that they are present any complaints or any incidents to report about queens and drones and failure of the colony. I would like to hear that, because we know that it's happening, and we've been discussing this with Jeff Pettis. So if somebody is thinking about it, please. Yes. No, you can do your discussion or your point.
Let me make some remarks. I do not refer to St. Chanta, I do refer to Mr. Peter Campbell. He told us that, he wanted to show us that there's full transparency in the approval with the studies and all this stuff. So that's the opposite of my experience there in the practice. So in Austria, we have a law. It's legal. It's a legal case where all every inhabitant of Austria has the right to know all the information on environmental relative studies also. Yeah? Okay. If we go to our authority and say, okay, there's a approval process for the neonates. Okay, so then we are allowed to make a presentation of the situation, but it was not in 2009. We had problems with losses of bees in active season, not just winter. We were not talking about winter. We are talking about losses also in the active season for 5 years. So since 2008. So last year, the content of the presentation was till now now we did not have any damage in corn. So as beekeeper, I asked them, hey guys, that can't be. The only damage that the Diabrotica made is our dead bees. That cannot be. And that points out one important thing. The aim, the approach of plant growing is to have 100% yield. So to get this, there is a lot of chemicals brought out to get this, yes? And for me, we are talking not on a bee problem, we are talking about a plant growing problem. So you can, but it's not fair. You cannot solve your problem in the pocket of your neighbor. And that's what, as beekeeper, for me, it's yes, no problems. There was not one single plant of corn which had a problem with Diabrotica, but only the dead bees. The next thing, what I think, because always the question is of field, how to get congruent of laboratory and field results. In my opinion, we have we have to complain the loss of the no impact zero group, because there is you won't find any field where is no impact. It's correct. It's a multiple causes, yes. But if you want to make a field study, we know that the bees forage for kilometers. So how you want to make a zero group, zero impact group? So and what happens? I have to be cautious to not to say too much. But if you make a trial and you want to show the impact of pesticides. So there's no obvious impact. But at the same time, at the control group, there is damage of dead bees. So what do you do? Is this no impact observed? Because even the control group, which is not a zero group, has problems. And if you want to make yes statistics, there's no effect you observe, and that's in my opinion it's also a big problem. So yes, that's it.
Okay, thank you. One moment. As for the citrus, for example, for citrus plantations, I do know there are alternatives to neonics, and they are biological control, but they are quite cheap. So, yes, very short comment because it's his turn.
5 seconds.
5 seconds. 5 seconds again. Okay, 5 seconds. I count.
5 Italian seconds.
I count.
10 minuti.
You're already
Bayer per ore. 10 minuti.
10. No, 10. 5.
Bayer parla.
Come on, Piotr.
Singenta parla e i ricercatori tacciono. Ma cos'è questa storia?
Come on, Piotr.
So, can I summarize the last two contributions regarding the study described by Peter Campbell? We have a very good study with high statistical power because many replicates. And the study, unfortunately, the results say there is no statistical significant difference between treated and untreated. And it doesn't mean this treatment is bee safe, because we have 2 hectare plots, and normally bees in an agricultural context fly over 700 hectares. So in those particular exposure conditions, there is no statistical difference between the bees placed in an untreated area and bees placed near 2 hectare plots with treated plants. That's all. This doesn't mean the treatment was bee safe, I think. Thank you.
Okay, Valde.
This is exactly my point. I was in the middle of this neonic battle and looked at a lot of studies, and one of the problems was that a lot of the studies had the problem that in our agricultural landscape, with the pervasiveness of the use of neonics, it seems to be close to impossible to have a neonic-free control in your field trial, and if your study design isn't capable of figuring out whether your control was actually neonicotinoid free, then it's actually useless. And that's why when we're talking about what to do in the next 2 years, because of soil persistence, okay, even if we want to do before and after comparisons, unless we very carefully design the studies to figure out whether we ended up running into a soil persistence problem or not, those comparisons will also be very difficult. The other point was the buffering effects of the honeybee colony, and therefore being a very difficult subject to study. I think when you look at the title of the EFSA guidelines, what is very encouraging in there is it talks about honeybees and bumblebees, and basically honeybees are not the only protection goal, and therefore to get an answer to certain questions, we can actually maybe do some of the tests with organisms where you have a little bit less complexity. And my final point is we should remember how we got to this point. The tests for approval supposedly showed safety and the products were approved. Okay, then they were just used, nobody else looked at this anymore. Then the beekeepers in the field made observations and says there's something going on here, okay? When the beekeepers went then to industry and said there seems to be something going on with your product, the industry said, oh, this is just in the field, we need lab tests, okay? Now, years later, we have a ton of lab tests, and the industry says, oh, this is great, but what about the field, okay? And in my mind, we have kind of a global field test, because I'm not old enough to judge this, but I don't think we have, we have all kinds of problematic insecticides before, but I don't think in the history of beekeeping, we had the beekeeping community globally up in arms, okay? And every time I was told by industry that, oh, in this other country they have no problem, okay? Sure enough, because I'm a little bit visible in the beekeeping community, people from that country said, can you come to us? We have this problem here, okay? And actually, I think there's also a duty for industry. There was the point from Canada, okay? Why are we running into these problems? And we're now finding out that the Italians had already studied this before, okay? And you are the global player, and instead of taking the experience from one country and saying, okay, there was a problem, we need to fix it globally, we have the exact opposite. We actually need the beekeepers network, okay, to communicate what's going on from one country, and there's an attempt to tell the Austrians, even in Europe, that something is going on in Germany, where we have to tell them, gee, this is not as true, they didn't tell you the truth about that. Okay, so I think we have a global field test that is already telling us we have a serious problem. We need to, the industry asked for the lab test, they got them. There was a very good point that we need to pay attention to the lab test. And I think the document that EFSA presented with the new guidance documents, it's not perfect, like Josef said, you want 100% yield on your crop, and we should accept 7% loss, on the bee side. That's also in the document, so it's definitely not perfect. But a lot of lessons were learned from the neonicotinoid battle, and I think we might make a big step forward if industry wouldn't challenge the new EFSA guidelines. Thank you.
Thank you, Walter. We have 2 more statements. Yes? And the lady there after. Yes, this is the lady.
Yeah, thanks a lot.
Okay, another one, but then afterwards.
Yes. Actually, I met a girl last year. She was working in a famous agrochemical industry in Belgium. And she told me about a project that they had of using the bee as a transport for pesticides. So in order actually to avoid pollution of the soil. So my question is really, I'm not a specialist in pesticides, but I'm a human, and here I cannot really understand how companies can put money in these types of projects, and it was quite big plants projects. And they know that they have several problems with bees concerning pesticide. I cannot really understand where the money goes there. I think it's a question of priority. Okay, maybe you'll say I'm a bit humanistic, but I am. Another thing is that this girl, she was talking about that, and she was convinced that it was really the better way to use this pesticide. So here there is a problem, I think, of objectivity. Okay, I don't know what is your feeling about that. Thanks to answer.
I mean, I'm not really...
Probably no answers anymore, please, because we have to finish it. Keep it short, and Francesco, you're the last one. Keep it very short, please.
Okay, I'll try my very best. I'm Melanie von Orloff from Germany, from a national protection organization called NABU. And actually, when I'm asked by the press, what do you think about the decline of the bees? First, I'm asking, which bees you're referring to? Because actually we honey beekeepers, I'm a beekeeper myself, we are really in a quite good status. We may lose a hive or 2 or so, but the drama is going out there. If you look at the decline of the bumblebees, of the social bees, of the social wasps, it's incredible. There's a big, as Walter said, a big field trial ongoing, and latest publication like Teneke's book about disaster in the making is linking all those pesticides with the decline of water birds, for example, because of the leaking of those pesticides into the water. And so I think there's one very simple solution for all the problem and all this argument about field test and lab test. Very simple. We stop using the stuff for the next 20 years, not 2 years, and see what happens. And usually, if I'm right, hopefully, the bees will be better afterwards. We'll see.
Well, thank you very much. Francesco. Okay. Yes, it is the last one. And then we will...
J'ai une petite question. Si l'intérêt, la transparence, l'intérêt public de vous, c'est le même de Marlboro, Philips, de l'industrie de la dioxine, de British Petroleum dans le golfe des États-Unis, qui vient de l'industrie atomique au Japon. Vous êtes de même niveau, vous êtes le pire.
I've got a question for you from for the transparency engagement. Have you got the same set of engagement as Marlboro, Philip Morris and the tobacco industry? The same level of engagement as the oil industry, and the dioxin producers in the industry, the amiante producers.
Oh, amiante. Non, c'est bon. Des petroleum mais il y a une autre question qui n'a pas ici été évoquée. On a parlé d'améliorer les méthodes scientifiques. Et vous refusez parce que ce n'est pas votre intérêt.
And there's another point here is that we spoke here about getting in better sets of testing, of scientific testing, and you refused.
Mais il faut améliorer la capacité publique de gérer le conflit d'intérêts par les mêmes qui font le contrôle.
There's a need to increase public funding and to avoid conflicts in interest in expert groups for the future.
Madame Alix de Novartis, qui après a fait Syngenta, est passée à faire le contrôle public en France. Et en France, l'unique enrobage autorisé, c'était de Syngenta pour le maïs.
Miss Alix from Novartis that came to the French body for authorization of pesticides obtained in France that just one product was agreed, that's a product from Syngenta, Cruiser.
Madame Thompson et Madame Alix s'est passée après à DuPont. Ça, c'est un bon contrôle public des intérêts privés. Hein?
Same for Mrs. Thompson. Mrs. Alix then came back to private industry, and this is a good public control over industry.
I remind you, we have only 7 minutes until the time we have.
Non, c'est une petite histoire. Madame Thompson, après avoir produit une étude qui a été certifiée par EFSA comme infondée, et c'était bien placé cette étude comme temps.
Ms. Thompson that has produced a study that has been stated by EFSA as unfunded, yeah.
A été prise directement comme employé directement, évident de Syngenta.
Has been used as an evidence by Syngenta.
Donc il y a un problème d'améliorer et de contrôler le conflit d'intérêts. Et ça, c'est vraiment un problème de la société, là où il y a des grands intérêts et des petits sujets, ou bien des sujets qui sont de toute la société. Et ça, c'est un point fondamental que je crois que Apimondia, il doit se charger.
There's an important point that has to be taken by Apimondia is the interest of small, weak, let's say, beekeepers or the public interest in comparison with strong private interests has to be taken into account and avoid this kind of conflict of interest made by people going from one side to another of being in private than in public.
Je termine seulement que j'ai tout le droit de me méfier complètement de votre transparence et parce que je sais que vous avez des intérêts et vous défendez des intérêts et c'est la vie.
And I've got to state that I've got the right to be cautious about your results, industrial results, because you've got private interests, and that's your right to have these interests.
Et ce qui est démontré que la science agronomique, et moi je suis heureux d'être agriculteur, a déjà l'alternative à vos pesticides mortels.
And I want to state that I'm a farmer, and the farming industry has already got alternatives to your killing products.
La rotation de culture, c'est pas convenable parce que vous ne vendez pas. Mais pour Diabrotica en Italie, la rotation de culture, ça marche beaucoup mieux que toute la merde qu'on jette dans la terre.
Crop rotation works. You don't support it because you don't sell it, but it works in Italy better than all the shit you are selling.
Thank you.
Thank you. Okay, so we can conclude.
Yes.
Because
Thank you. Thank you. All these hours now, from 2:30 almost 4 hours, we heard about the effects of the studies, we heard about the laboratory and the field studies, how we can go from one to another, what do we need. We heard about the interaction, not meant, metabolites, phytoremediation, need for more studies for funding. We actually heard about almost everything of the points of discussion that we set up in the beginning. And we would like to conclude them in a kind of a text that we will read. Gilles will read it for you, and it will be used as a draft for the declaration of Apimondia.
the draft of the draft. Do you hear me? No. Yeah. Okay. The draft of the draft, because we don't have lots of time. The idea is for Apimondia, is to go further, because for the last 116 years, at the end of each Congress, we have resolutions. Apimondia recommends this, Apimondia is against this, Apimondia wishes this, etc. And nothing happened between two Congresses 2 years. So we want to go further and to not only print our resolutions in our proceedings books or CD-ROMs or websites, but we want fundamental text about our fundamental problems we are facing. Not only the pesticides, but also adulterations, the cost of the new criteria for laboratory analysis, etc. We have plenty of problems. Of course, this one, the bee mortality, it's the most important one. How an average of 34–36% of colony losses, for example, in the last 5 years in USA. Imagine the reaction if it's the same in the cow industry or the cow farming. It will be a big scandal worldwide. So we cannot divide the surviving cows in two. We can divide the colonies in two. It's why it's not so shiny, but it's a real problem in terms of economy for our beekeepers. A real problem. I must tell the scientific representing the industry. Okay? So, it's not that we are against chemical products. We use chemical products in our beehives, okay? But when they are killing our bees, we are against this. The text could be something like Apimondia supports completely, but we will have an answer because it's on the process of the vote. So we still sorry to be contacted to be against what you said, but we still doesn't know the final decision, except if you know how heavy is your lobby, and we can say that we support, if it's adopted, we support it. If it's not adopted, we support the text of EFSA proposition. And not only we recommend this as an example, as a material for all the other countries outside of European Union, in terms to have a globalization of the harmonizations. We talk about harmonizations, and I think that everybody in this hall is agree with this issue. And you're right. Your writing is very. So we also not only support this kind of proposition, but we can go further, and we can include more things, like even if it's at the moment unrealistic economically or in terms of laboratory, etc. But we want to include the metabolites. We want to include the multiple expositions, and even the remnants of the soil. We don't have real studies about this, independent studies about this. And another recommendation in this topic of pesticide is to have independent funding for long-term studies in a large scale. Studies. Okay. What?
And for ring tests.
Yeah.
Ring test.
Yes.
For ring tests as well. Yes.
And honey.
Yeah. Yes. And we must also all together work on something about the seed dressing, that we have automatically systemic action. Is it good or not good? Because before it was easy to spray pesticides before the blossom period or after. With this kind of diffusion in the nature, it's more difficult for us. And to go in a positive way, and just not to say no, we are against this, etc. Of course, Apimondia supports the eco farming, etc., etc. Because it's a future. We are going to the wall. We are at the moment we have products 7,000 more toxic than the famous and prohibited DDT. And in the next generation, it will be not a supplemental effect with 0.01 ppb, but 0.0001. Where we are going like that? We are going straight to the wall. We are fighting the nature. We must work with the nature. And the eco farming is not to go back 60 years ago or 70 years ago, when we used very, very few pesticides in the nature. It's exactly the opposite. In 70 years, we have lots and lots of new knowledge about what is going on the nature, the crops, etc., etc. And yes, the synergies between pesticides, etc., it's important. We must also include in this kind of resolution and fundamental text, it will be one part and the other part adulteration, etc., that the beekeeping sector must have a better representation. When you said 80 people from the industry and 3 from beekeeping, and we are talking about beekeeping, it's strange. It's unfair. And of course, we must be connected with the other species of bees, solitary bees, etc., bambus, and it's a very good approach from EFSA, and we must go further. And at the end, we must conclude that everything we are saying must be can be realistic if we are out of conflict of interest. If we can work without the lobbying pressure, and lots of things like that, with the deputies, with people in charge of decision, with the toxicology commission, etc., etc. We must have something independent. Okay, that it's a global thing, and we will include it to a document, and this document, it will not be signed and diffused tomorrow at the closing ceremony because we don't have time. But we will work on it. We will spray to all of our members, and when we will get the last suggestion, we will send it to all the UN agencies, all the ministries of agriculture worldwide, 196 countries, and a maximum of media, of course. Because until now, when we are talking, it's between us and okay, we have nice papers on our different beekeeping magazine. We must go further and we must inform all the world about our problems and not saying, oh, we want money, we want to support, we want succeed. No, we want to propose nice propositions, positive approach and alternatives to this war against the nature.
I would like to thank all the speakers for the patience and active participation. I would like to thank you for being here and congratulate you for your patience because it's really late. Thank you very much.
And I want to thank you, Fanny Agina, for the moderation of this. So I will say everything.
Sorry? I think you don't think my subject is more stretches. Keep it up to field,
yeah, but
and then we see afterwards, because all the field and lab test debate.
You know, they do it by the book. They will drag this into journals. They will be fighting my science against your science, and, until they find something, the new kind of thing that will bring them revenue. So we start studying this thing from the ground up.
2 years is just not enough time to change the things. I fear that they will debate and debate over 2 years.
But this is the strategy actually.
Yeah, and that's the point. And these 2 years will be used by several companies to feed the politicians and the deciding sites the information they want them to
Can
know.
you stage some protests over there? Because at least from a beekeeper's perspective, I think this is the only thing that we can do actually to contribute to it. Because we cannot get into this debate. Obviously, we don't have the knowledge and the authority, but I think we should press on with protests of some sort.
I see. I'm always glad to see that there's so many people involved in that, and also the research. It was very interesting here in Apimondia to see all those different countries doing research in this field and providing all the needed information. I mean, even if there's one lab test or 2 or 3 not in the right way, all of them can be failures. There's so many of them.
Yeah, but it's not the matter if they succeed because they go and fight it with another paper. Maybe it's fake or maybe it's not, but...
Yeah, so far there haven't been published so many papers from the point of view of Syngenta and so on. So far, I mean, starting now with their first paper is actually really a good idea that they start to publish their own stuff. This gives at least something, some sort of arena where you can start to argue around. Now you have the data. Before that, they haven't even ever published anything. They always keep telling, we are checking, we are proving, we have all the data, and nobody sees the data. That's complicated. It's really complicated. Those guys are brave.
Yeah. Have to give them that.
Yeah.